Good morning, ladies and gentlemen. Please take your seats and welcome the President of the American Epilepsy Society, Dr. Manisha Patel.
Good morning, everyone. I am very pleased to welcome you to the Presidential Symposium at the 77th meeting of the American Epilepsy Society. I greatly look forward to us all sharing knowledge and discovery to the ultimate betterment of and care of people with epilepsy and to the renewal and development of lifelong friendships that is the hallmark of this meeting and our society. Before we start our program, AES wants to acknowledge that we are on the Miccosukee, Seminole, and Timucuan land. The Miccosukee tribe of Indians of Florida and their Seminole and Timucuan relatives have lived in Florida since time immemorial. The Miccosukee and Seminole people held on to the southernmost front through fierce resistance over half a century of guerrilla warfare with the U.S. military, while most Timucuans were removed to Cuba by the Spanish Empire a century prior. Orlando's history has a non-native settlement dates back to Fort Gatlin, built in 1838 during the Second Seminole War, when the Miccosukee and Seminole were being pushed out of the Central Florida Reservation into the Everglades, where they now live today. We say, Shon Ha-Besh, thank you to the Miccosukee and Seminole people for continuing to set an example of political autonomy and cultural preservation in Florida. Next, I would like to take a moment and remember Dr. Jun Atsushi Wada, Professor Emeritus in the Department of Psychiatry and Neurosciences at the University of British Columbia, who passed away this year at his home in Vancouver, Canada. He was president of AES in 1989, president of the American Clinical Neurophysiology Society in 1985, co-founder of the Canadian League Against Epilepsy and its president from 1977 to 1979, and an officer of the Order of Canada. He had just celebrated his 99th birthday. Doctors Nico Moshe and Jerome Engel wrote a heartfelt obituary, so I'd like to read from that. They call Dr. Wada a luminary and guiding light for generations of epileptologists and basic scientists he mentored, and a longtime colleague and friend, and also an innovative pioneer in translational epilepsy research and human brain asymmetries. They said he projected an authoritative but always humble demeanor and epitomized the well-rounded scholar. His interest in art and humanity included writing, painting, and poetry, and was a role model for all of us. He greatly appreciated his adoptive homeland of Canada and was known for enclosing maple leaves in his letters. A note card painted by Dr. Wada, shared by Dr. Moshe, include an inspirational message for all of us here. The future is all yours for the creation of new knowledge and the exploration of the brain. This extraordinary meeting, of course, does not happen without tremendous energy and committed work by many. I especially want to thank Dr. Barbara Jobst, Chair of the Council of Education, Dr. Ignacio Valencia, Chair of the Annual Meeting Committee, and Dr. Jennifer Kearney, Chair of the Scientific Program Committee. Please join me in recognizing their efforts. And listed here are the names of the outstanding group of member volunteers who have directed the major symposia and the investigators' workshops for this meeting. Thank you. And now I'd like to take an opportunity to ask all new AES members in the audience to rise so you can be welcomed. "2,000 new members joined the society this year to date, and our membership now stands at over 5,100. I want to thank you all for being part of this vibrant, growing professional community. I'd also like to ask those of you who are attending the annual meeting for the first time to please stand, and welcome to all of you. And one final ask: if you are not a new member or a first-time attendee and you happen to be seated next to one of these new colleagues, please take a moment right now to introduce yourself. We are a welcoming community, and it starts right here. Thank you. Now it is my special honor to recognize a group of individuals, our newest members of the Fellows of the American Epilepsy Society class. This is the 8th class of the American Epilepsy Society Fellows, our peer-to-peer recognition of professional accomplishment and dedication to the epilepsy and to AES. I want to personally applaud our new 2023 FAES class of 78 members, and we ask all the members in attendance to please rise. Our fellows are now 669 strong. Thank you. Please rise if you're a fellow of the Americans. The AES meeting is indeed a worldwide gathering. We have nearly 1,100 attendees with us from 70 countries. AES is proud to be a chapter under North America of the International League Against Epilepsy. I am honored to introduce Dr. J. Helen Cross, President of ILAE, to present an update.
Thank you very much, and thank you to the American Epilepsy Society for giving me this opportunity to provide a very brief update about the current issues we face within the International League Against Epilepsy. My disclosures. So I'm just going to give a very brief overview of who we are, for those of you who may not know, and then just outline where we are with 3 particular challenges that are currently facing us. Not least, implementation of the Intersectorial Global Action Plan to help close treatment, inclusion and research gaps in epilepsy worldwide, addressing the needs of people with epilepsy during crises, and the overall impact of climate change and trying to reduce the impact of those we treat with epilepsy. So what about the International League Against Epilepsy? Well, we are the global organisation representing the professionals worldwide involved in the care of those with epilepsy. We have 129 country chapters, but overall, bearing in mind that some chapters include more than one country, for example, the Caribbean, we have a presence in excess of 160 countries and territories worldwide. And as Dr. Patel has just outlined, the American Epilepsy Society is one chapter, country chapter of the International League Against Epilepsy, one of our largest chapters. This just gives a brief overview of the structure and how we work. We have an executive committee elected with a 4-year term, of which we have subcommittees. We then divide into regions, and actually chairs of each individual regional board are members of that executive committee. We have councils for continuity of key aspects of our strategic plan, and then what we call commissions looking at individual topics moving forward, and special task forces. But what I want to highlight from this is the increasing sections that we now have, which are for individuals of like interests. Young Epilepsy section has been steadily growing, now up to membership of almost 2,000. We now have an extremely large nursing section, approaching 1,000 in its membership. We also have a very rapidly growing technology section, pharmacy section, and soon to be launched a social work and social services section, available for anybody with those interests to join. Of course, education is one of our key missions, and our academy now has the curricula complete for primary care, level 1 and level 2, with resources also available. And we move towards outlining resources with regard to how the level 3 or advanced stage of a curricula will be implemented. But we have also many regional education programs that are, of course, targeted at those regional needs. And we've launched an e-forum series that has been ongoing since the pandemic, utilising webinars and ongoing microlearnings. And just to update with regard to our 3 publications, Epilepsia, Epilepsia Open, and our education journal, Epileptic Disorders, and their renewed impact factors. And we thank our editors for the hard work they undertake in order to maintain their excellence. We also have moved forward with our communication. We have podcasts that have been extremely successful. I actually understand they're in the top 15% of downloads from Spotify, which is fantastic. Then we also have various YouTube videos, various social media platforms to continue to communicate what we do. We launched a strategic plan in 2021, and then came IGAP, which was a 10-year plan for epilepsy and other neurological disorders. We have been we have made specific targets with regard to what we want to achieve over the over the 10-year period. 90, 80, 70. 90% of all people with epilepsy being aware of their diagnosis as a treatable brain disorder, 80% of people with epilepsy having access to the appropriate and affordable, safe anti-seizure medicines, and ultimately 70% of those treated achieving adequate seizure control. And with the launch of the Intersectoral Global Action Plan on Epilepsy and Other Neurological Disorders, approved at the World Health Assembly in May 2022, it gives us a real tool to work towards that. This global agreement was approved by all 192 member states of the World Health Assembly, which commits to those member states moving forward with what's drafted, and indeed with regard to review as to what progress has been made. And in parallel, an epilepsy technical brief has actually been published with support from the International League Against Epilepsy, looking at levers and tools that can be utilised for change. We see this as a key to unlock political momentum. It has 5 strategic objectives, 4 of which are general to neurological disorders. And the final one, number 5, is very specific to epilepsy. And within those 5 objectives, there are 10 targets. And this is what these 10 targets look like for epilepsy, all aiming towards closing the treatment gap, closing the inclusion gap, and then ultimately closing the research gap, with a target of doubling research in each individual country with regard to epilepsy and other neurological disorders. We've started by trying to get information worldwide about where we are, where we stand with each of these individual targets. And you can see, although there seems to be some areas in which we seem to be already making good progress, unfortunately, orange and red on this heat map mean that there's very little information and we're nowhere near the targets currently. We have provided an acronym, and indeed various tools will be provided for individual countries to move forward with each of these targets, not least looking at engagement, plans and information, access, care pathways, looking at providing a standardised care pathway, training and education also being integral to how we move forward with this. Of course, we've all become very aware of recurrent crises as have been seen around the world over the past few years, and many of them are ongoing as we stand here today. And it became quite clear to us that we really need to look at the needs of people with epilepsy and provide support where we can. This map is obviously constantly changing, but it demonstrates that even though for certain crises may be in the fore of our news that we see day to day, there are many, whether it be natural, whether it be man-made disasters, that are ongoing and which need support worldwide. So we've set up a crisis task force, and what this is trying to do is just liaise with humanitarian groups to make sure that we have the right medications, the right support going in when these disasters happen, but also providing information that's readily available both for clinicians and people with epilepsy and in multiple languages. And finally, none of us can be unaware of the fact that climate change probably is a reality. And maybe how is that going to affect us, and how can we support those around us as it moves forward? So we actually have a climate change commission who are trying to look at what we do, make sure that we're compliant with various needs, and also how we can support those with epilepsy. What we are doing in a major way is combining forces with other organisations, and particularly with the Intersectoral Global Action Plan, the International Bureau for Epilepsy, the Patient Organisation, the World Federation for Neurology, the International Child Neurology Association, and of course the WHO. And together over the next few years, we want to really move forward, engage with different countries and their ministries of health, and really move forward to implementing the Intersectoral Global Action Plan. Thank you very much.
Thank you, Dr. Cross, for that ILAE update and for all the outstanding work of the ILAE on behalf of epilepsy. At this time, I want to recognize the companies that support AES through medical education grants, charitable grants, sponsorships, exhibiting, and advertising. I invite the representatives from these companies to stand as I read your company's name. At the very highest level of support, UCB Incorporated. This is the benefactor level. Thank you to our leader level supporter, Jazz Pharmaceuticals. And then I will read a list of partner level support: Acadia Pharmaceuticals Incorporated, Catalyst Pharmaceuticals Incorporated, Livanova, Marinus Pharmaceuticals, Neurelis Pharmaceuticals, SK Life Sciences Incorporated, and Takeda Pharmaceuticals. Please stand if you're in the audience. At the supporter level, we have BioCodex, Biohaven Pharmaceutical, NeuroPace Incorporated, Sumitomo Pharma America, formerly Sunovion, Upshur Smith Laboratories, and Xenon Pharmaceuticals. Thank you. As a reminder, please take time to visit the exhibit hall in the West Hall C, Level 2. It's an important part of our meeting and a great place to learn from the best and latest information from our industry colleagues. And now, it is my pleasure to start the Presidential Symposium. I'd like to acknowledge that this symposium is supported in part by an educational grant from Livanova. And also I'd like to introduce my co-chair, Dr. Dan Lowenstein. Dan Lowenstein is Robert B. and Eleanor Aird Professor of Neurology and Executive Vice Chancellor and Provost of the University of California, San Francisco. Dr. Lowenstein takes care of patients with epilepsy and also conducts basic and translational and clinical research. He is an ardent advocate for patient groups as well as for epilepsy researchers and clinicians. And importantly, he was past president of the American Epilepsy Society and is a wonderful mentor to all of us in the society. He and I are very honored to chair this 2023 Presidential Symposium. And now I'd like to introduce a very special award, starting with an introduction of Dr. Fritz Dreyfus. The Fritz Dreyfus Lecture honors the memory of Dr. Fritz E. Dreyfus, who was a leading clinical epilepsy specialist, clinical investigator, and former president of the American Epilepsy Society and the International League Against Epilepsy. Dr. Dreyfus was born in Dresden, Germany, and escaped with his family in the 1930s, moving first to South Africa and then to New Zealand. He received his medical degree from the University of Otago in New Zealand, completed graduate studies at the National Hospital, Queen Square in London, and joined the faculty of the University of Virginia in 1959. In the mid-1960s, he began working on a collaborative project in epilepsy with the NIH. His pioneering work in objective documentation of Epson's advanced clinical investigation in the epilepsies as a whole is well recognized. Dr. Dreyfus was founder of the Comprehensive Epilepsy Program at the University of Virginia. He was mentor to a generation of epilepsy researchers and an educator with worldwide influence. As a member and chair of the ILAE Commission on Classification and Terminology, he tackled a job most thought impossible. And through all, he remained devoted to caring for patients, including clinics in underserved areas of Southwest Virginia, which continue to this day. His influence has touched nearly everyone practicing or studying in the field. And now, it is my sincere pleasure to introduce Dr. Kelly G. Knapp, MD, FAES, who will present the 2023 Fritz E. Dreyfus Lecture. So I'm going to introduce Dr. Knapp's qualifications and accomplishments first. Dr. Knapp is an associate professor of pediatrics and neurology at the University of Colorado. She is my dear colleague and collaborator. She received her medical degree from the University of New Mexico School of Medicine, completed her residency in pediatrics at Children's Hospital of New York, followed by pediatric neurology residency at Columbia University at Children's Hospital of New York. After her residency, she trained as a clinical fellow in pediatric epilepsy at the Columbia Comprehensive Epilepsy Center at New York Presbyterian Hospital. Dr. Knapp now practices at Children's Hospital Colorado in Aurora, Colorado, and is the director of the Dravet program and interim section chief. Her interests are epileptic encephalopathies, including Dravet Syndrome and infantile spasms. She is a founding member of the Pediatric Epilepsy Research Consortium and continues to serve on its steering committee. This group focuses on developing collaborative research across the country for children with epileptic encephalopathies. Dr. Knapp serves on the board of directors of the American Epilepsy Society and on the medical advisory boards of the Dravet Syndrome Foundation, Coalition to Cure CHD2, and the Scientific Advisory Council of Cure Epilepsy. It is my pleasure to present Dr. Kelly G. Knapp as the 2023 Dreyfus Lecturer. Kelly, please join me at the podium here to receive your award. We will begin our session with an introduction, followed by Dr. Knapp's lecture. So now we start the Presidential Symposium at the Cutting Edge Epilepsy Therapies in 2023 and beyond. Can we bring up Kelly Knapp's slides? Thank you. Or actually, slides number 1, speaker. Thank you. So we start this lecture with Dr. Knapp's lecture, A View from the Clinic, A Need for Better Treatments. And I just want to give you an overview of our goals for this symposia. Our goals for the symposia were to look at the road beyond and understand where we are going from where we are in 2023. And so Dr. Knapp is going to start us off with this view from the clinic and how we need better treatments for epilepsy.
Thank you, Manisha. And boy, there are a lot of people out there. First, I want to say thank you for this named lecture. It's such an honor. And I would be remiss if I didn't acknowledge all of the people who've helped me get here, because this has not been work that I've done solely on my own. All of the work I do is in collaboration, and there's so many people I collaborate with. I'm not going to begin to name names because I will leave somebody out, and I don't want to offend anybody. But thank you to all of you who collaborate with me in clinical trials, observational trials, and all of the other work that we've done, especially my colleagues through the Pediatric Epilepsy Research Consortium. I also want to just take a moment to acknowledge my mentors, particularly Amy Brooks-Kayal, who I'm sure is here somewhere in the sea of people. Amy saw something in me that I didn't see in myself and has created many opportunities and support for me, and for that I am eternally grateful. I also have a whole list of other mentors along the way, and I'm sure I'm going to leave somebody out, but I'm going to name a couple of people. Bill Gayard, Tracy Glauser, Ann Berg, Dennis DeLugos, thank you for answering my phone calls and my emails and answering them the second and third and fourth and 12th time. I appreciate it. My peers have also been great support to me, and thank you for that. And most importantly, to my family who is here today. I wouldn't have been able to do any of this without you. Thank you for being honest with me when I have not gotten work-life balance quite correct and supporting me anyways. Thank you so much. So with that, my task this morning is to describe where we are in clinic today and make an argument for a need for better treatment. I'm sure that all of you would probably agree with that statement. So my task is easy, but thank you for sitting through this anyways. I do have disclosures. I do participate in research and consulting and safety monitoring boards. I think the most important thing to acknowledge is that I'm a pediatric epileptologist, so what I present today will be through that lens. So for the adult epileptologists out there, thank you for your patience. I thought I'd start off with a typical day in clinic. I live in Colorado, and we cover a really broad area. And a typical patient for us is presented here, an 18-month-old of Latina and Native American descent who had a recent unprovoked generalized tonic-clonic seizure and a history of a prolonged febrile seizure in the past. She has already had an MRI and a normal EEG, and she comes to us from Silverton, Colorado. And for those of you who are not familiar with Colorado, Silverton is in the southwest corner, and it's about an 8-hour drive to get from Silverton to Denver. There is flights that can be taken, but they're quite expensive. And to get from Silverton to Denver, you have to go over 6 or 7 mountain passes, depending on the route that you take. So this is very challenging for families to get to us. And I think you guys will be familiar with this list of questions. I think this is what I hear from many of my patients in clinic. Exactly how many people have epilepsy? I've never heard about it before. Why does my loved one have epilepsy? Will this medication that you're recommending work? How will this change our life now and in the future? Will my child be able to drive? Do I need to keep saving money for college? Will she be able to attend college? And why does a seizure happen anyways? So I'm going to walk through and answer some of these questions and talk about what our needs are in our current patient population. So answering how many people develop epilepsy is pretty straightforward. This really hasn't changed significantly over time. When we look at the incidence of epilepsy, these are the new cases of epilepsy in a given period of time. About 61 people per 100,000 person years worldwide will have epilepsy. If we look at the prevalence of how many people have epilepsy now, it's 6 per 1,000 people worldwide. If we take the population of the world, and this is my math, which I think I did correctly, we end up with 50 million people with epilepsy right now. That's an incredible amount of people. There's perhaps been an increase in epilepsy over time, and this is data that comes from the Global Burden of Disease that was funded by the Gates Foundation. They specifically looked at idiopathic epilepsy, which they identified as genetic and unidentified causes of epilepsy. So this excludes some percentage of people with epilepsy. And from 1990 to 2016, they found a 6% increase in people with epilepsy in the prevalence, so the people who had epilepsy at that time. This is perhaps due to people living longer. If you look a little higher up in the column, you'll see that death rates have actually decreased. So death rates from epilepsy have gone down by 24% in that time period. So we definitely have made some success there. And so that may account for some of this increase. And I think we're all familiar with this graph on the bottom right, where we see the the prevalence of epilepsy across the ages, where we have an increase in the younger years, and then pretty flat throughout the middle of life, and then increasing again in the elderly. These are things that we're pretty familiar with. Why does my loved one have epilepsy? Well, I think we've all come to realize that epilepsy is really the epilepsies. There's lots of reasons why people have epilepsy, and seizures are often just a symptom of the underlying disorder. Epilepsies that used to be considered cryptogenic or idiopathic are now better understood as genetic. And even with clear underlying imaging abnormalities, there may be genetic underpinnings. A parent of a child with epilepsy famously said, "My child's disease is a flight number." I actually think they look a little bit more like license plates. A lot of what I see in pediatric epilepsy is genetic epilepsy. This is a slide that gets put up all the time. Ingo should have charged us for it. But we can see that there's a dramatic increase in the number of genetic diagnoses that are attributed to epilepsy. And in fact, it's very difficult to update this slide because the curve is so steep. Ingo tells me we would need to have a slide that goes 3 or 5 slides up in order to get them all on. So let's circle back to our patient. As a reminder, this is a patient who had a prolonged febrile seizure and then had an unprovoked generalized tonic-clonic seizure, had a normal MRI and a normal EEG. And so we did the next thing we know to do, which is genetic testing. And as we often find, this patient had 3 variants of unknown significance. So now what do? We still can't really tell this family why their child has epilepsy. I think it's important to acknowledge that epilepsy syndrome is often not specific enough, and as many people know, one of my areas of specialty is infantile spasms, so I'm going to use an example of a typical 5-month-old with infantile epileptic spasm syndrome. If that child has an etiology of CDKL5 deficiency disorder, we know that they will have a poor response rate to even standard medications. When we look at outcomes at 14 days, children with CDKL5 deficiency disorder will have a response rate of 26%, whereas a national population has a response rate of around 58%. And if we look a little further out, things are even more grim. For this patient population, at 3 months, their response rate is 4% compared to 53% in the national infantile spasms population. If this child has a different disorder like tuberous sclerosis complex, we know that response rates to vigabatrin will be much greater. And in fact, vigabatrin may have even prevented or delayed the evolution into infantile spasms. And this does raise the possibility of, can we prevent other epilepsy syndromes when we have an early diagnosis? And I think this is work that we have left yet to do, but I think something that many of us think about when we're sitting in clinic on a day-to-day basis. So epilepsy really is a collection of rare diseases. We have some official definitions of what a rare disease is. In the U.S., the Orphan Drug Act defines a rare disease as something that impacts less than 200,000 people. In the European Union, there's an official definition of a rare disease of something that affects less than 1 in 2,000 people. Well, we know that epilepsy in the US impacts greater than 3 million people, so it doesn't really meet our definition for a rare disease in the US. But when we start to look at specific syndromes or etiologic reasons for epilepsy, we can see very quickly that we meet definitions for rare epilepsy. So epilepsy really is a collection of rare diseases, and I think many of us are starting to think of it this way and tailoring our approaches. And we've had lots of discussion about precision medicine, which addresses these rare epilepsies in this way. I think it's important to recognize that there are a lot of barriers to studying rare diseases, and we are making great progress in this area. But when we have rare diseases, it's challenging to do a randomized trial, which means we end up reporting case reports and case series. When I report this from my institution and you report this from your institution and somebody else reports it from their institution, we end up with 10 different ways that we should be treating this patient, which makes it really challenging for us to agree on what to do in a randomized trial, because it's hard to randomize to 10 different arms. And then there's other factors that we need to consider. When we're looking at rare diseases, because treatment may only impact a small number of people, the cost of treatment can be quite expensive. It may not be fully accessible everywhere. Parents are fearful of participating in randomized trials, and we're making great progress in this area, but we need to make more. We may not even have the appropriate diagnosis or a timely diagnosis allowing us to intervene at appropriate times. These are barriers that we're working our way through, but it's important to acknowledge that we have them. Rare disease requires collaboration. We need to make sure we have consistent diagnosis, consistent treatment approaches. We have a couple of different ways that we can establish this, and we've seen increased use of a Delphi process to assist with this so that we can get expert opinion out there. The International League Against Epilepsy is working to establish consistent definitions so that we're, from site to site, we're making the same diagnosis and using the same definitions. We have a number of natural history studies that are helping us as well. There are numerous examples of observational studies, many of which are prospective, which are particularly helpful. And we have some efforts with the Epilepsy Learning Health System and the Pediatric Epilepsy Learning Health System that are helping us so that we have a good idea of what natural history looks like and what we're actually doing now for these patients. And we do have clinical trials that address treatment, but often it's necessary to involve multiple centers, and so that requires significant collaboration, which we are getting very, very good at. The next question that many families ask is, what is the likelihood that this medication will work? And I think most of us historically report that 30 to 40% of people living with epilepsy will still have drug-resistant epilepsy. So 60 to 70% of people do respond to medication, but we have this large percentage that doesn't. The most recent prevalence data on this sits at 30 to 32% of people with drug-resistant epilepsy, so we really haven't made significant change in this population. And quite frankly, this is who I see in clinic. I don't often get to see that 60 to 70% who respond to medication. They're seen by my general child neurology colleagues. We have a great idea that any treatment should really have no seizures and no side effects. And I think that we're able to attain this in a large percentage of our patients, that group that respond to medications. And we are getting closer to having better and better side effects with our medication. But overall, we have little change in response rates, and we still have this group that has drug-resistant epilepsy. And we're always in that particular group balancing seizures and quality of life. And so for my patients, I think a more realistic goal is the least amount of seizures possible with the best quality of life possible. While we hope for no seizures and no side effects, that's not a realistic goal for many of the patients that I see, but we sure do celebrate it when we get there. We have so many medications from which to choose from. I think this is a slide that everybody puts in many of their talks, and we see this steep development of the treatment options that we have. And I think that this is leading to better side effects, but again, we still have the same amount of population that is not responding. Quan and Brody had this landmark article, and I apologize, I thought I had fixed the typo on the slide. This is from 2000, not from 2020 in the New England Journal, where we saw success of anti-epileptic drugs, and by the time you get to the third medication, the response rate is around 4% when we look at populations of people. Repeating this work in 2018, these numbers look exactly the same. I'm really not sure what the statistical difference is between 45.7% response to a first medication and a 47% response to a first medication. When we look at the difference between new medications and old medications, this is a study comparing lacosamide with carbamazepine, and we can, lacosamide's in the blue and carbamazepine's in the green, and we can see that there's very little difference in response rates to old medications versus newer medications. In the top left is looking at all seizures. In the top right is looking at focal seizures. On the bottom left is looking at generalized tonic-clonic seizures. And then in the bottom right is looking at an elderly population, people greater than 65. And that's really the only place where we start to see a little bit of difference. So really, it may not matter whether we choose an old medication or a new medication from response rate, although it probably does impact side effect profile. Fortunately, we have other treatment options, and we're going to hear a little bit later in the symposium some of the treatment options and some of the exciting therapeutic things that can happen with stimulation. I love this picture that shows all of the stimulation options that we have readily available now, including VNS, responsive neurostimulator, and deep brain stimulators. And we're seeing increased indications for these. I will say my experience in my patients and when we look in the literature is that we definitely see a reduction in seizures. We see that this can help with side effects. Seizure freedom is not very common with these devices, and so we're still not, while we're improving potentially quality of life for our patients with drug-resistant epilepsy, it's not very often that we're seeing seizure freedom with these treatment options, although we can have increased personalization of treatments for our patients. Epilepsy surgery, I think, is not something that we're talking too much about in this symposium, but there's lots of other presentations about epilepsy surgery throughout the meeting. And certainly this is an effective approach to treatment. This is just one study demonstrating the higher response rate of surgery in the pediatric population compared to medical management at 7%. And we also see better scores on the child behavior checklist, the PEDS quality of life, and Vineland measures. So epilepsy surgery is something for my patient population when we have candidates is something that we really do need to be considering. Yet there's a lot of literature out there that families are afraid, providers are afraid of doing this. I think we are all well aware that patients aren't getting referred to epilepsy centers to be evaluated for epilepsy surgery. And access to care can be quite limited, and we have some special populations that access of care seems to be more limited. Access to good communication and knowledge is essential. When we think back to our patient today, who at this point is probably not a surgical candidate, but having to drive 8 hours over 7 mountain passes to come back to get neuropsych testing, do an EMU admission, get a PET scan, a SPECT scan, and talk to us multiple times about epilepsy surgery is quite a burden for that patient, and we need to wrap our heads around how to better do that. Gene modulatory therapies are currently in trials, and these are super exciting, particularly for the patient populations that they will impact. But these are very specialized treatments to very specialized populations, and is not going to help probably our broader group of drug-resistant epilepsy patients. And we'll be hearing more about this later in the symposium as well. So super exciting and super helpful for patients that they impact, but it is not going to impact our larger group of patients. We are talking more and more about health and equity, and I think that this is an area where we really need to focus what we're doing in clinic right now today. We know that we see increased rates of uncontrolled epilepsy across many, many studies for some of our patients. Black adults have more uncontrolled epilepsy than white and Hispanic adults. Non-whites have a higher incidence of status epilepticus. The lifetime prevalence is 2 times higher for Black persons. We see more ED visits for non-whites. We have lower medication adherence for Black patients, and we have the lowest rates of neurology follow-up for American Indians and American Natives, such as the patient that we're talking about today. This is an area where we need to figure out how to make improvements. These are patients that we're seeing today and now, and I think we need to stop documenting how inequitable our care is, and we need to figure out how to intervene and change this. We see increased use of phenytoin compared to levetiracetam, and we also have lower rates of surgery in Black patients. These are areas where I think we can start acting now. Shifting gears a little bit, quality of life is a major issue for our patients with drug-resistant epilepsy, and there are a handful of manuscripts documenting this, and we're starting to hear patient voice, and I think it's really important that we recognize this. This is looking at the Lennox-Gastaut population, and we can see that these patients are impacted not just with seizures, but with sleep disturbance, injuries from their seizures, severe developmental delay, mobility issues, educational issues, communication issues. And parents are able to prioritize these things for us. Seizures and safety often are the first thing that they're concerned about, but close second are issues with communication and behavior, as well as other morbidities. And we need to make sure that we're keeping track of this as we're thinking about our therapeutic options for these patients. We are all well aware that retailers are really good at using real-world data to target us and to help us in making our choices when we're on Amazon or shopping at Target or getting flyers in the mail. And we need to be better at this. But it turns out it's not so simple. I have yet to see a patient who comes in with a simple barcode on them that allows me to put them in a box. So a little bit different than buying a box of Band-Aids. The pediatric epilepsy learning healthcare system is starting to look at this, and they actually have some ongoing projects looking at second-line therapy for neonatal seizures, the use of oxcarbazepine and risk for infantile spasms, and prediction of epilepsy after neonatal seizures. But it still takes time to mine this data and sort through it, and so we need to get a little faster at this. We also have the Epilepsy Learning Health System, which is making great strides, and but I think it demonstrates some of the challenges of pulling data out of our medical records. So this is a study that was just recently published looking at 3 factors that we should all be documenting every day on our patients, including seizure frequency, ILAE classification of seizures, and date of last seizure. And when they asked people how well they were documenting this, I think people were pretty honest, and they thought they were pretty good about seizure frequency, not so great about ILAE classification and date of last seizure. It turned out when they actually looked at objective data in the medical record, they were worse than they reported. So I think that's something that we all probably need to carefully take a look at. And it makes it really hard to use this data if we don't have great data. They tried 2 different interventions, one with a flow sheet that actually increased the documentation to 100%. An intervention without a flow sheet was actually worse than what people were doing already. I think those of us who participate in research and collect data in databases know how important this is. We don't leave free text options in research databases, and we probably need to be a lot better in our clinical documentation if we want to pull out data in being able to put some of this data in boxes so that it's more easily coded and better able to be pulled out. There's a lot of interest in the Epilepsy Learning Health System, and there's been great gains in pulling out data. There are 9 sites contributing provider-reported data. There are 8 sites reporting patient-reported data. There is more work to happen to get all of the sites on board. It takes a lot to take data out of the electronic medical record and get it into a database. So there's still a lot of questions that I'm not able to answer for my patient. How does a seizure start? I don't think that we know the answer to that. What medication will work best for me and for my patient, not just people who are similar to me? That's really challenging to answer. And quite frankly, for every one of our patients, I do not have a crystal ball, and it really is an N-of-1 study that we're doing with each one of our patients. And that's frustrating to our patients as well. Do people with an earlier diagnosis do better in the long run? I think we're starting to get some data to say that that is probably true, but we need more of that. Will AI and imaging make things better for me in clinic and for our patients? I'm hoping so, but I think we have yet to see. Is driving important since we will have self-driving cars in the future? I get asked this question by almost every single one of my patients' families, and I don't know the answer to that. I'm not sure when self-driving cars will come. Maybe Dan will have a view on that. So what is the impact on our clinical care? I think we have so many treatment options, and it really is great that we have all of these options, and we're allowed to partner with our patients and families, and that is more important than ever so that we can talk about what the best option will be for that individual patient. We do need more approaches than just anti-seizure medications, and we certainly have those, but we haven't made a lot of progress in our drug-resistant epilepsy patients, so there's a lot of room for improvement still. We need to be able to learn from our current practice. We have a wealth of information in our electronic medical records, and we are making progress in figuring out how to pull that out, but it turns out that that is a really big project that we need to keep chipping away at. Health inequities contribute to some portion of our failure to attain no seizures and no side effects, and I think each one of us needs to be aware of this and think about how we can get around some of these barriers so that all of our patients have access to the best care that they can receive. I think there's a lot of great information coming up, and I'm looking forward to see what the view from the clinic may be 20 or 30 years from now. Thank you.
Thank you, Dr. Knapp, for that wonderful Dreyfus lecture. And we're going to continue on to our main program. I just want to remind everyone that this educational activity is supported in part by an educational grant from Jazz Pharmaceuticals. And now I'd like to move on to the next part of the program. If you could. So for my end, I don't have any disclosures to report. And I just want to give you a learning objective from our entire group here. We want to be able to describe the rationale and mechanistic basis of therapies that are in the pipeline now and apply this knowledge in choice of therapy by balancing the benefits and risks, and also integrate the knowledge from new therapeutic approach into the future clinical care of patients. So this is what the whole scope of our presidential symposium is. As you've heard from Dr. Knapp, where are we currently in clinical care for epilepsy patients? But now we're going to start hearing about what are the new cutting-edge therapies in development. And then Dr. Lowenstein is going to really close our symposium by telling us about the clinical visit of the future and what that might look like. And throughout our talks, as you heard from Dr. Knapp, there's going to be an incorporation of health inequities in each of these presentations, where will patients have equitable access to future therapies. So I hope you can make these conclusions as well. So we've heard from Dr. Knapp, who's really set the stage. And next, Dr. Lowenstein is going to introduce Dr. Gemma Carville.
Thank you, Dr. Patel, and I'm pleased to introduce Dr. Gemma Carville. Dr. Carville is an assistant professor in the Department of Neurology at Northwestern University, and her lab uses genomic technologies and computational tools to define the molecular basis of epilepsy. This includes both coding and non-coding variants and the development of machine learning algorithms and high-throughput functional assays to distinguish between disease-causing variants and benign genetic variation. Her research also integrates epigenetics to build models of non-coding regulatory elements that control gene expression and uses these approaches to understand pathogenic mechanisms in patient-derived stem cell models. Welcome, Dr. Carville.
Thank you so much, Dan, for that introduction. Everybody who knows me knows I love to talk about genetics, so I'm very excited to be here. But I think what's even more exciting is that 10 years ago, when I was one of those folks who stood up at their first AES meeting as a new AES member, I think I could never imagine that 10 years later, I'd be standing here telling you not only about the amazing progress that we've made in terms of understanding the genetic basis of epilepsy, but to be telling you about preclinical treatments. I think I could never have imagined that, and this is very exciting to give you an overview of kind of the state of the field and where we're at, as well as the challenges that are facing us in terms of recognition of these gene-targeting therapies. And so to start, I have the following disclosures in terms of consultants and speaker engagements. I feel like I should follow Kelly's lead and say my other disclosures that, of course, I am a geneticist, and so all of my talk will be through the lens of being a geneticist. And so these are our learning objectives, so you can read them for yourselves, but really 3 main points that I want to drive home is that I really want you to walk away from this understanding that the pathogenic mechanisms that underpin the genetic epilepsies are really going to dictate which of the gene therapy options we're going to use in particular cases. And then also to understand the breadth of epilepsies that are potentially treatable by gene targeting therapies in the future. And so this is an updated slide, somewhat updated slide from Ingo's. This was made by my very brave graduate students, and there are definitely some genes that are left off here. But I think this gives a nice overview of the evolution of genetics in epilepsy. And so really when I started in the field, roughly 10 years ago, the genetics of epilepsy was largely thought to be due to channelopathies that were very rare and mostly limited to syndromic cases. And this has really changed over the last 10 years or so, where exome sequencing by many small groups, larger consortia, have really changed the way we think about genetics in epilepsy, and there are now hundreds of genes that we know to be associated with this condition. And I like to think now we've kind of moved into what I call a gene matcher era, where now, rather than groups on their own making gene discoveries, it's now very much a worldwide collaborative way in which we identify those very, very rare genetic causes that are present in a handful of individuals. And so, with this rapid progress in terms of the identification of the genes that cause epilepsy, we now know that at least in the developmental and epileptic encephalopathies, the largest cause of these disorders is through monogenic causes. So those genes that I showed you, it's mostly de novo, recessive, highly penetrant variants, where a single variant or 2 variants in the case of recessive are sufficient to cause disease. And that really has been, at least initially, where we made the most progress in terms of genetics. We also now, however, appreciate that both the genetic generalized epilepsies and the focal epilepsies, there are a couple of rare monogenic cases, but overall there is probably a much more complex genetic architecture. Things like polygenic risk scores are coming more and more to the fore, where it's a combination of multiple genetic variants that then predispose you to developing GGE or focal epilepsy. We also know in the focal epilepsies that somatic mutations can also make a large contribution to the development of epilepsy. And so clearly my title is Gene Targeting Therapies, so I'm going to talk a lot about targeting these specific genes in the genetic epilepsies. But I think one of the other things I hope to leave you with is that targeting the abundance of specific genes and proteins is likely to be broadly applicable. So here I'd like to disagree with Kelly respectfully, that I think that it probably will not only be those rare monogenic epilepsies that we can target through gene targeting mechanisms. And we can have a debate at the panel later as to who believes who. All right, so first off, what are gene targeting therapies? So this is a throwback so that we're all on the same page in terms of how genes are expressed and how they work. So what I'm showing you here is a promoter region, here is a gene of interest, and so when a promoter region is active, that means that the gene is being actively transcribed and you have RNA molecules that are produced. And so in the case of a monogenic epilepsy, what we would then have is this change of an A to a G. And so this would be a pathogenic variant or a mutation that would then lead to that monogenic epilepsy. And so it would be present in the DNA molecule. and then when this DNA molecule is actively transcribed, it would be present in this case in about 50% of the RNA molecules, and then that would lead to an effect on the protein. And so when we talk about gene targeting therapies, what I'm really talking about is targeting at either the DNA level or at the RNA level to correct or address this particular pathogenic variant. And so there are a variety of different ways in which we can do this. One of them is what we call gene editing therapy, and that would be where we would correct back the mutated G allele back to an A allele. And so then essentially what would happen, this should disappear, is that this would be now only the G, which is the reference allele, and there would be no pathogenic RNA that was made. And so gene editing therapy is very exciting. I think in epilepsy, we're a long, long way away from recognizing this technology in our field. I think maybe in 5 to 10 years, and in some other conditions, there have been exciting advances in terms of gene editing therapy. So what I'm going to focus more on today are a number of other approaches that are in development in the epilepsies. And so one of them is gene replacement therapy. And so this is essentially where we take the coding sequence for a particular gene and we insert it into the genome. And there are a variety of different ways in which we can do this. And in general, when we insert the gene into the genome, there tends to be more than one copy in the genome, which is important to keep in mind for later. So this is gene replacement therapy. Another place where there is a lot of excitement in the field is with antisense oligonucleotides. And so this is when we create short fragments of DNA that can bind specific RNA molecules, and depending on where they bind and how they act and their specific targets, we can use these antisense oligonucleotides, or ASOs, to either increase or decrease the levels of RNA. Another approach that I'm going to talk about also involves either increasing or decreasing the amount of RNA for a specific gene, and this is through CRISPR-guided activation or inactivation. And so this is where we have what we call a dead Cas9. So Cas9 normally cuts DNA when it's guided to a specific sequence. But if you have a dead Cas9, it now is only merely recruited to the region you want, in this case the promoter, and we can couple it to an activator or a repressor. And then we can either increase or decrease the amount of RNA for that specific gene. And so, as I mentioned, we can use these approaches to either increase or decrease the amount of RNA and protein. And the choice we make as to which way to go really depends on the pathogenic mechanism that underpins that particular genetic condition. And so I like to use this example of the sodium channels. So SCN1A and SCN2A are two genes where de novo variants cause the developmental in the DEEs. And so SCN1A, loss of function mutations, mean that there is therefore less sodium moving through this channel. This means that when we use things like sodium channel blockers, this can actually make the phenotype worse because now there's even less sodium moving through the channel. Conversely, something like SCN2A can be caused by gain of function mutations. And so here there is an excess of sodium coming into the cell. And so in these instances, sodium channel blockers can be an effective treatment. And so taking the same paradigm, when we think about how do we target these genes, what's really important to keep in mind is that with loss of function, what we're trying to do is increase the amount of protein, whereas with something like SCN2A, we would want to decrease the amount of protein. And so I'm going to give you two real-life examples of how folks in the field have done this. So the first is an ASO that was targeted to the gene SCN2A. So remember, now we have a gain of function, and we want to reduce the amount of RNA. And so to do this, we use RNA-H mediated degradation of the mRNA, where we design an ASO to the gene of interest, so in this case, SCN2A. And because what ends up happening is there is an RNA-DNA hybrid, this signals RNase H to come to that molecule and essentially degrade that molecule. And so you have less SCN2A. And so what this particular group did was they used the mouse model for SCN2A, so that's a very specific missense mutation here that is found in many patients with SCN2A. And so in this particular mouse model, they used an ASO targeting SCN2A. And you can see very nicely, sorry, I'm just going to move things around here. You can see very nicely here in the control ASOs, this is the level of SCN2A, and this is a reduction in the mice that were treated with the SCN2A ASO. So along with this reduction of SCN2A, what the group observed was that this SCN2A knockdown causes rescues early lethality. So both given at a dose at P1, so this is the control, here we have the treated mice, and we can see that there's a decrease in early lethality. And this was extended even more with 2 doses. And importantly, because in general, we will only be able to treat patients, presumably, after seizure onset, they also treated the mice at P14, and they also saw a rescue of the early lethality phenotype. They also saw that there was a rescue of the seizure phenotype at P40 to P45, but interestingly, not later. And so when they looked at the levels of SCN2A at these later time points, what they found was that essentially the ASO washes out or is degraded over time, and there is a return to baseline, and that correlates with the then increase in seizures. And so that's an example of knocking down a protein using an antisense oligonucleotide, but we can also increase gene levels using ASOs. And so this is an example of SCN1A, so you remember that is due to a loss of function model. And so the way in which this works is by taking advantage of a very nifty system within the cell that is naturally occurring. And these are poison exons. So poison exons are short exons that when they are included within a particular transcript, it triggers a nonsense-mediated decay. And so you can see in this example, when the exon is skipped, you get the full-length protein, whereas when the exon is included, this PTC is included and the transcript is degraded. And so importantly, these poison exons are naturally occurring and present in about 30% of genes in the genome, including in SCN1A. And so a putative therapy is that if you can design an ASO to block the inclusion of this poison exon, you can actually bias the machinery to create the full-length and fully functional protein. And so Laurie Isom's group at the University of Michigan tested this approach in the SCN1A mouse model. And what they found was that, I'm showing you here the non-productive or the SCN1A transcript that contains the poison exon. And so with the ASO, you can see a reduction in that transcript. And concomitantly, you can see an increase in the transcript that codes for the full-length protein. Along with that, you can also see an increase in the amount of protein. So with an increase in protein in these mouse models, there was both a reduction in seizures compared to untreated or PBS-treated mice, and there was also an increase in terms of or a decrease rather, in the early lethality. And so this, again, is a nice example of how we can use antisense oligonucleotides to increase the amount of protein in loss of function or haploinsufficient conditions. And so these are 2 examples where we know what the gene is. It's a genetic defect that causes the epilepsy. But I hope to convince you in the next couple of slides that it's not only the genetic epilepsies that we could potentially treat by targeting specific genes and proteins in the genome. And so this is a very beautiful example from Dimitri Kuhlmann and his group, and it's an example of gene replacement therapy. And so now we're not talking about antisense oligonucleotides anymore. Now we're talking about inserting a specific gene and a promoter, so the part that drives the expression of the gene, into the genome. And so normally what folks would do, they would just put in a promoter that is constitutively active, so active in most cells. It would just always be expressing this gene. But you may not always want this. And so the beauty of this study was that they devised a system where this particular gene would only be active in instances of increased neuronal activity. And so to do this, they put the therapeutic gene under the control of an immediate early gene. So at normal physiological conditions, this gene is not expressed. Increase in pathological neuronal activity, this promoter now switches on, and the gene and the protein is made, and that will restore normal physiological activity. And so to test this hypothesis, what they did was, again, with the CFOS promoter, so this is an immediate early gene, so they put the gene KCNA1, which encodes a potassium channel, under the control of the CFOS promoter and had a GFP so that they could visualize. They then used a chemoconvulsant model of epilepsy, so kainate injection to induce status epilepticus in the mouse, and then injected both control and the KCNA1 gene protein under the control of the CFOS promoter into the mice. And so what you're looking at here is 7 different mice and the number of seizures that each mouse is having. And so this is our control, and so no KCNA1. And you can see that both before and after, there are a large number of seizures in each individual mouse. Conversely, in those mice that were treated with the AAV containing the KCNA1, you can see with each individual mouse, there is a reduction in the seizures, which can then also be collectively quantified. And so here there's a reduction in the amount, a reduction in seizure activity with increased KCNA1 expression. And so this is a chemoconvulsant model where it's, I think, a beautiful example of how we can use activity-dependent promoters and specific genes and proteins to rescue seizure phenotypes in mice. Staying with the KCNA1 gene as a potential target for upregulation in epilepsy. In addition to just replacing the gene, another approach that several groups are using, including Gaia Colasante's group, is to use CRISPR-A. So here you'll remember, this is where we use that dead Cas9 version, the dead version of Cas9, so it now no longer cleaves the DNA, but it's coupled to this activator. And so we use a single guide RNA to guide this particular construct to the promoter region of KCNA1. And so when it is bound here, you get an increased expression of KCNA1 and increased protein levels. And so very similar to the previous study, they also used the kainate model to introduce this particular guide RNA. They show an increase in KCNA1 expression and protein levels. And very similar to the previous study, you can see again each of these lines is an individual mouse pre and post treatment. And very similar to before, what was shown is that in the presence of the upregulation of the KCNA1, there is a reduction in seizure presentation. And so most of what I've told you so far is very happy and rosy, and I think it's very exciting. The number of tools that we have, the number of groups that are trying to do this, and the very promising preclinical data that we have. But I also would be remiss to not acknowledge the challenges that we have in the implementation of these technologies in human patients. And so one example that I want to give is an example from our own work where we spend a lot of time thinking about gene dosage and the amount of protein present. And so the gene CHD2, when there are de novo loss of functions in this particular gene, it causes developmental and epileptic encephalopathy. So again, haploinsufficiency for CHD2, a DEE phenotype. But through an international collaboration, what we identified recently is now 3 individuals who have a deletion of the upstream long non-coding RNA called chaser. And these individuals present with a somewhat more severe phenotype than those with haploinsufficiency for CHD2. So they present with frontal lobe atrophy, hypomyelination, and in general, these individuals are nonverbal and non-ambulatory. And so, of course, one of our major questions was, what is chaser doing? Because it sits right in front of the CHD2 gene in terms of expression of CHD2. And so it turns out that when you delete chaser, this actually leads to an increase in the amount of CHD2 protein. And so really what I'm highlighting here is that both too little of the CHD2 protein as well as too much of the CHD2 protein is very much detrimental to neurodevelopment and neurological function. And so I think this is my Goldilocks example, that both too much and too little is probably bad. And this is not just for CHD2. There are a number of other genes that we know they are dosage sensitive, and that loss and gain of function phenotypes, loss of function mechanisms, are associated with neurological phenotypes. And so I think we need to be very thoughtful in terms of how do we very finely tune the amount of protein that we're either reducing or increasing in a lot of these genetic epilepsies, and then with ramifications for other types of epilepsies as well, where the genetic predisposition may not be as strong. And so that's just one example of a challenge, but I think that there are many. This is a circuitous diagram that shows our path on the way to cures. And I've shown it as one path, but it really could be hundreds. But I couldn't figure out how to make a pretty slide with hundreds of railway tracks and still get the content across. So just use your imaginations. But I think some of our challenges really revolve around timing. When do we treat? Is after the seizures too late? And I think there's a lot of questions about when to treat. I think one of the other challenges that we have is that many of what I've shown you today, those genetic epilepsies have great animal models. They very nicely recapitulate the clinical phenotype that we see in patients. But this is not the case with the overwhelming majority of genetic epilepsies. So I think we need to be exploring other models, both animal as well as cell models. I think iPSCs are exciting, but they are very much still an emerging technology. Things like delivery, if they are non-integrating, then we're going to need likely repeated dosing. And then there are concerns with integrating as well. Clinical trial design, Kelly very nicely touched on some of these points, and I think we need to be thoughtful around this as well. And then lastly, I think the other thing that we really need to be thinking about is ensuring equitable access. I've shown you that very nice diagram of how the genetic epilepsies, we pretty much know that every DEE, and for about 80%, we know what the genetic cause is. That's true in most high-income countries. In low and middle-income countries, individuals can't even get genetic testing. And so I think we need to be cognizant of this not only in America, but across the world as well. And I think we need to be thinking about cost and access as well. And so with those downers and all the challenges that we have, I do think we still have a lot of opportunities for targeting genes, not only in the genetic etiologies, but also broadly across epilepsy, so that we can treat the cause, but not the symptoms, and be a cure and not a treatment. And so I think this is a very exciting time for the field, and I'm excited to see what the next 10 years look like.
Dr. Carville, for that excellent presentation. I'd like to introduce Dr. Farah Lubin. Dr. Lubin is Distinguished and Endowed Professor in the Department of Neurobiology at UAB. She directs the NINDS Neuroscience Roadmap Scholars Graduate Program and several other training programs. She's an epigenetics researcher and heads the Neuroepigenetics and Memory Laboratory at UAB. And her lab is focused on developing cures and identifying mechanisms that are evident with epigenetic mechanisms so that we can improve memory and better seizure control in epilepsy. Welcome, Farah.
Thank you, Manisha. I have no conflicts or disclosures, really. So the learning objectives for my talk today is to have you appreciate and understand that transcriptional reprogramming occurs in epilepsy, specifically with epilepsy development. To have you understand that the role of epigenetic and subsequently metabolic regulation of epigenetic mechanisms are involved in regulating these transcriptional programs involved in epilepsy, and how we're currently exploring how we can manipulate epigenetic mechanisms in order to treat certain aspects of epilepsy. And then hopefully identify certain barriers that we can overcome in order to apply epigenetic therapy towards the treatment of epilepsies. So here I'm showing you a timeline of epileptogenesis in regards to acquired epilepsies. And we appreciate that this process involves three main phases: the insult phase, where there's an initial trigger leading to prolonged seizure activity or status. And then there's this latency period where there's no seizures presented, and then followed by chronic unprovoked reoccurring seizures. And what we do appreciate and understand is that during these three phases, there's multiple changes occurring. First, with abnormal regulation of a number of ion channels that we've appreciated over the years and understand, this regulation of immediate early genes, and then this latency period is not as quiet as we initially thought, right? There's a number of transcriptional events occurring that results in this restructuring of many circuits within the epileptic brain that gives rise to this progression of epilepsy and chronic seizures. And so when we think about transcriptional programs that are occurring in epilepsy development, we have to realize that these transcriptional programs are playing a role in various aspects of the disease, including seizure generation, as well as disease progression. And so here, as a molecular geneticist, I'm going to talk about dots, and these dots are going to be genes. And so here I'm showing you a volcano plot where we've actually mapped what genes are being affected in response to first the acute stage of epileptogenesis. And so in blue, we have the genes that are being downregulated. In red are genes that are being upregulated. And by looking at the pattern of regulation of these genes over time, you can see that they are changing. And so what's responsible for all of these transcriptional gene regulation occurring during the different phases of epileptogenesis, and how can we explain how these genes are being affected? So my lab and that of others have hypothesized that there must be a larger mechanism regulating and coordinating the regulation of these genes. And so we've turned to the science of epigenetics to explain how we are having long-lasting and persistent structural changes contributing to the development of epilepsy and its progression. And so when we think about epigenetic mechanisms, I want you to truly appreciate what's occurring within the micronucleus. We know that DNA is a 3D structure, and it needs to be unfolded to allow certain factors to activate and silence genes. And so when we're thinking about regulation of gene transcription, you have to recognize and appreciate that there's the unfolding of chromatin that needs to occur. And this process is highly coordinated by a series of writers. These are enzymes that recognize certain areas of chromatin, the proteins, and add various post-translational modifications. And these writers with these post-translational modifications help to recruit what we call readers. And these are proteins that recognize these marks and help to assemble larger complexes to regulate transcriptional activation and silencing. And then you have the erasers. So these are the enzymes that come in and remove these marks. So this is a tightly regulated process, but it's totally dysregulated when it comes to insult and leading to epilepsy. So whenever I try to describe what it means to have transcriptional reprogramming and chromatin remodeling that's contributing to that, I take you back to Bio 101, where we first studied and maybe were introduced to the idea of molecular genetics. And that is that DNA is transcribed into RNA, RNA is translated into proteins, and proteins make up the very synapses that we often study when we think about circuitry restructuring in the brain and how these circuits are dysregulated in the context of epilepsy. And we now appreciate that they are both genetic and non-genetic factors that contribute to this abnormal dysregulation that we're seeing and observing in epilepsy. And so the science of epigenetics is just a little over 15 years old, in that we are now appreciating that these mechanisms that were once thought to only be established when in terms of determining cellular phenotype are very much active and remain active in response to our environment, in response to insults, in response to abnormal genetic processing, and that they are contributing to a number of functions in the brain, including synaptic plasticity, epilepsy, memory formation, aging, neurodegenerative disorders, psychiatric disorders, environmental exposures on brain function, and so on. But we know very little about how epigenetic mechanisms are regulating transcriptional programs in the context of epilepsy. And so can the science of epigenetics give us answers? And I say yes. So I'm going to give you several examples of how we are currently looking at epigenetic modifications and their contribution to epilepsy. So first example is that of histone modifications. So here I'm showing you a variety of modifications which include histone methylation, phosphorylation, and acetylation. And they can, depending on which lysine residues and which histone proteins associated with DNA, can result in the opening of chromatin, allowing for gene transcription, or the closing of chromatin, resulting in gene silencing. And so the example I'm showing here is of two post-translational histone modifications, one of which is the dimethylation at K4, which is responsible for transcriptional activation. And you can see here that in control animals compared to our epileptic animals, which is the pilocarpine animal, there's a decrease in this activation mark. There's also increase in a repressive mark called the K9 dimethylation mark. So what this suggests is that with these marks being both active and repressive, you're getting up and down regulation of specific genes that are associated with these modifications. So not only have we determined this in the pilocarpine model, but it's also been demonstrated in other acquired forms of TLE rodent models. This has been demonstrated to definitely be involved in downstream of a number of signaling pathways that are disrupted with epilepsy, specifically within the hippocampus. We've demonstrated that these marks are very much involved in regulation of a number of genes that are dysregulated and contributing to epilepsy. We've demonstrated that the erasers are very useful at employing and reversing some of these marks. So histone deacetylases have been shown to play a major role in the development of temporal lobe epilepsy, but that if we manipulate these marks, and I'll talk about that in terms of epigenetic therapies, if we manipulate these marks, we can reduce seizures and improve certain comorbidities. In addition, we've demonstrated that not only is this relevant to rodent models, but it's also relevant to human patients. And so my lab and that of others, we have an opportunity to work with resected tissue, and we have validated these marks are indeed involved in epilepsy. Another important mark in regards to epigenetic regulation of gene transcriptional programs are DNA methylation. We know that DNA methylation plays a very important role in transcriptional silencing, and that this balance of regulation of transcriptional silencing and activation is significantly disrupted in epilepsy, and a number of gene regions have demonstrated to be disrupted in regards to their DNA methylation patterns. So an example of that is here, this heat map. Just pay attention to the colors. So the red colors here indicate low levels of methylation, and yellow colors indicating high levels of methylation. And just looking at just controls versus epileptic, this is epileptic human patients or TL from the hippocampus. And you can see there's a shift in the methylation patterns that occurs across multiple genes. If you were to just take two genes, for example, we have the CAMP kinase 2 gene and the interleukin 10 gene, and you can see based on dots, that's the number of cytosines that are being methylated. So you can see that with, for example, with CAMP K2 controls versus epileptic, there's an increase in number of cytosines that are being methylated. And in the case of interleukin 10, you see that there's a decrease in the number of cytosines that are being methylated. And this corresponds to an inverse relationship in regards to gene expression, where you can see that when you have a gain of DNA methylation, there's a reduction in gene expression. When you have a loss of DNA methylation, you have an increase in gene expression. So we've demonstrated this to be the case across a number of genes important in the regulation of brain function and disrupted in epilepsy, and that's with BDNF, brain-derived neurotrophic factor, and receptors such as GRIN2B, the NMDA glutamate receptor subunit. We've demonstrated that gene expression is changed across multiple genes expressing for inflammatory proteins, including interleukins. We've demonstrated that the pattern of DNA methylation occurring at gene regions is very different in regards to status epilepticus, which is prolonged seizure activity, and then very much altered in the basic recesses of the epileptic brain. And excitingly, manipulating these DNA methylation marks and changing or reversing transcriptional reprogramming has been demonstrated to be a potential treatment option for the development of epilepsy. Another epigenetic modification that we're very excited and interested in studying is non-coding RNAs. So for a long time, we thought that 98% of our DNA was non-coding, meaning not translated into proteins. We now appreciate that this is not just junk DNA, but there's very much activity involved in the regulation of these non-coding RNAs and how they are epigenetically modifying our gene expression. We now appreciate that our non-coding RNAs are acting as epigenetic factors, participating in the regulation of several pathophysiological processes involved in epilepsy, and that non-coding RNAs do indeed regulate genes that are involved in metabolic signaling and synaptic plasticity. So this is just to have you appreciate the world of RNA, non-coding RNAs. So we're typically used to our housekeeping RNAs, ribosomal RNAs, and transfer RNAs. But now we have to appreciate that these non-coding RNAs are very much active. And I'll give some examples of in which we're studying microRNAs, in that they are definitely involved in and active in and altered in human temporal lobe epilepsy. Several non-coding RNAs have been demonstrated to be involved in cell death that's involved in the process of epileptogenesis, and that by manipulating these long non-coding RNAs, we can dampen seizures. Also demonstrated is that some long non-coding RNAs are dysregulated and associated with comorbidities such as SUDEP, sudden unexpected death with epilepsy. And excitingly, these long non-coding RNAs can potentially serve as biomarkers in that they have been shown to profile the development of epilepsy, at least in rodents, and present very much in peripheral blood. And so they can serve potentially as biomarkers. And they are believed to be transferred through exosomes, which are basically vesicles from different brain regions that end up in our peripheral blood. When it comes to epigenetic mechanisms, we have to appreciate that metabolic signaling is very much involved in the regulation of these epigenetic modifications. So again, going to Bio 101, looking at glycolysis pathways, the TSA pathway or Krebs cycle, they produce cofactors that are absolutely necessary for these enzymes to mediate their post-translational modifications. So shown here, for example, is alpha-ketoglutarate, which is very much a byproduct or altered in ketogenic diet. And so, unfortunately, the molecular connection between metabolism and epigenetic modifications and its effect on chromatin remodeling is poorly understood in the context of epilepsy. And so what does it mean when we think about metabolic regulation of epigenetic mechanisms? So first, I want you to appreciate that temporal lobe glucose hypometabolism occurs in people with epilepsy. Temporal lobe epilepsy specifically, and so this is a PET scan showing here in the arrow showing glucose glucose analog FDG, and you can see that is the intensity is reduced, indicating that there's a loss of proper glucose metabolism. And so what does this actually mean when you think about the transcriptional regulation programs that are altered in epilepsy? And that what it suggests is that there are a number of metabolic signaling pathways that are disrupted. In addition, with changes in DNA methylation, these pathways are also targeted. So if you were to just look at the DNA methylation pattern of certain genes, so this is a gene ontology analysis, basically grouping genes into different pathways, metabolic pathways, and you can see that a number of metabolic pathways are impacted by just the change in DNA methylation, and shown here the same thing. So what this suggests is that epigenetic control of metabolic pathways are impacted in epilepsy. So what I've shown you so far is that epigenetic and metabolic signaling in epilepsy is altered, whether it's genome specifically to epilepsy or metabolism to epilepsy. We have to appreciate that there's an interaction or intersection in terms of epigenetics playing a role in both, or the convergence of genomics as well as metabolism on epilepsy. And so when we think about epigenetic therapy, we have to take all of these epigenetic modifications into consideration and appreciate that environment, such as nutrition, inflammation, stress, along with genetics and epigenetics intersect to create the epileptic phenotype. And so the goal of epigenetic therapies is to restore normal gene function, as normal as we can consider it, by resetting the switch that turns genes on and off, right, in terms of epigenetics. Unlike mutations in the DNA, epigenetic aberrations are potentially reversible. And epigenetic therapies offers a potential way to influence and manipulate gene transcription. So here, there's a number of ways we can employ epigenetic therapies. So we can nominate a specific modification that we see that's altered, depending on the type of epilepsy you're studying, and then employ a therapeutic or functional assessment of these modifications. We could change them through drugs, through dietary supplementation, or AAV delivery systems, or CRISPR-I, or activation, which we heard from Gemma, or even nanoparticles. So we know that there are a number of epigenetic therapeutic drugs that are already in existence, specifically because of the treatment of cancer. So we have DNA methylation inhibitors, where they can block these enzymes that mediate DNA methylation. We have histone deacetylase inhibitors, which I briefly mentioned, and these drugs target deacetylation enzymes to prevent the deacetylation of histones. And then with non-coding RNAs, we can employ AAVs in order to deliver these non-coding RNAs to specific targeted genes. And then when we think also about epigenetic therapy, we can consider dietary supplementation. So we know, for example, that the ketogenic diet is a high-fat diet and has been highly effective at reducing seizures in children and adults with epilepsy. But we know very little about the mechanisms by which the ketogenic diet works. But however, interestingly and excitingly, we know that the ketogenic diet can target a number of epigenetic mechanisms, including DNA methylation. And so if we could want to learn how we can employ or understand mechanisms of the ketogenic diet, an example here I'm showing is that of methyl supplementation, which engages the DNA methylation metabolic pathway. And we've demonstrated that we can inject methionine, which alters the methylation pathway, and that this results in an 8-fold increase in methionine in brains. And the idea is that methionine will then increase methylation at specific genes that are involved or have loss of function in terms of DNA methylation, and then reducing certain transcripts and silencing them. And we've done this in at least in the one epileptic model, the K8 model, where we've demonstrated that just changing the DNA methylation pattern through methyl supplementation is sufficient to rescue memory deficits in our epileptic rodents. We've done multiple tests. This is a fear conditioning test. Associative memory is altered with rodent models and in epileptic patients, but we are able to rescue that with methionine. Object location, which is another hippocampus-dependent task, is also rescued with methionine supplementation. So what these studies suggest is that just working with the methylation pathways and employing them at specific time points can potentially alleviate certain comorbidities. We heard also from Gemma about CRISPRs. In this way, we're employing CRISPR-Cas9 to manipulate specific genes or epigenetic modifiers to change the expression pattern at specific gene sites. So in this way, we can activate specific genes, recruit polymerase II, and increase and upregulate genes that are beneficial or reverse certain epileptic phenotypes. In terms of epigenetic therapy, we are looking and considering using nanoparticles. So nanoparticles are small particles that range from 1 to 100 nanometers in size. They are undetectable by the human eye. And the idea about nanoparticles is that we can package specific drugs. So one of the side effects of using a lot of these inhibitors I mentioned is that they have global effects. But if we can target them specifically to specific brain regions, then perhaps we can have more targeted and effective results. And so by packaging these drugs in nanoparticles, we can induce a focal ultrasound at a specific brain region and release their contents into the brain. So this is a great way of avoiding the blood-brain barrier, which it can also inhibit drug delivery. And so this is just after the focus ultrasound treatment where the nanoparticles, their contents have been released. So these results are really exciting, but the use of nanoparticles in terms of epigenetic therapy is still in its early stages of development, specifically in terms of where to target the focal ultrasound. We've been able to also deliver AAVs in this way, but more research is needed to determine the safety and effectiveness of this approach for different diseases, including epilepsy. So certain things that we have to consider when it comes to epigenetic therapy is cellular diversity. I like to call it unmixing the smoothie. So yes, we have a certain brain region, but we know that within those certain brain regions, we have different cell types that can differ in terms of their transcriptional profiles. And so using single nuclear RNA sequencing, we can begin to start looking at which epigenetic modifications are occurring in different cell types. So here we have so we have glia cells, we have inhibitory cells, different types of excitatory cells. And the point I want to make here is that you have different expression of different epigenetic modifiers in those cell types. And so we have to begin to understand how each cell type is different in their epigenetic makeup. This is just another way of looking at different cells and how they cluster and express for different levels of a specific enzyme. So lastly, I want to point out that race, sex matters when it comes to epigenetic mechanisms. So this is just an exploratory study that looked at race, ethnicity-associated blood DNA methylation differences between Japanese and European American women, clearly demonstrating that there's a difference in the DNA methylation pattern just based on race. And so we still need to do this in terms of males as well. So epigenetic mechanisms in the central nervous system, can they predict or prevent epilepsy and or associated comorbidities? The answer to this question is we have a lot of work ahead of us. Our understanding of the role of epigenetic mechanism in epilepsy is in its infancy. Epigenetic changes can be reversible, so it may be difficult to maintain the effects of epigenetic therapy over time. But with preclinical studies, we hope to better understand how epigenetic mechanisms are regulating these transcriptional programs, hopefully defining and designing better drug approaches or epigenetic therapies to manipulate them, and then going from the bench to the bedside for treatment. And so the impact on clinical care, epilepsy is one of the oldest diseases described in human literature and one of the most studied neurological diseases. Yet our approach to treating has remained unchanged for centuries. Part of the challenge is that the limited clinical classification of seizures is not reflective of the greater cellular and molecular heterogeneity underlying different seizure types. Epigenetic mechanisms underlying epilepsy has revealed that transcriptional reprogramming does occur, but very much in a very cell type specific manner. And we are beginning to link the functional significance of epigenetic mechanisms to the control of transcriptional programs in epilepsy and epilepsy-related comorbidities. So I look forward to the panel discussion and answering your any questions. Thank you.
Thank you, Dr. Lubin. So we're going to change gears a bit and move from omics to the cell and the network level. And with that, I'll introduce Dr. Robert Hunt, who is a faculty member in the Department of Anatomy and Neurobiology at UC Irvine. And his lab focuses on conditions of brain development and injury where a treatment does not yet exist. His group is creating new models of complex neurological disorders, developing cellular and molecular tools that enable precise manipulation of repair of the brain and recording high-speed dynamics of specialized neurons derived from human stem cells. Dr. Hunt.
Am I on? Okay. No, I'm not on.
And then I said,
Okay. Okay. All right, I'm all set up. I'm the only unique person with my own computer here. Being a little bit difficult. But thank you, Dan, for that kind introduction, and thank you, Manisha, for the invitation. I've been coming to this meeting for my entire career. I started as a first-year graduate student coming to this meeting, and it's really an honor to be here on the big stage with you all. And I'm very excited to talk about some of our work. I have no disclosures. I'm going to talk today about interneuron cell therapy and inhibition, and so the learning objectives center around that. Really, we want to understand some of the preclinical data supporting interneuron cell therapy. And towards the end, I'd like to cover some of the key differences between progenitor cells and stem cells as potential sources for cell transplantation. But before we get there, I really want to start in the 1800s, the 19th century. A lot of the concepts that we still think about today and study in the laboratory really originated in the late 19th century, and this is an example of this. Inhibition is no exception. In 1881, William Gowers, the British neurologist, was the first person to really conceptualize the modern understanding of inhibition in epilepsy. And Gowers was a contemporary of John Hewlings Jackson, and he had become convinced of Jackson's evidence that seizures start in the cortex. And Gowers reasoned that if that's the case, and seizures start from some overactivity in the cortex, and there must be some restraining force that limits neural activity. And he called this force a resistance or a type of inhibition. And he reasoned that if this is true, and if this is the case, and there's this overexcitation, and then there's this restraining resistance, that seizures and the discharge must result from some sort of a sudden decrease in this resistance, at least temporarily. And this was really a fantastic insight that he had 70 years before the discovery of GABA. And so this is amazing insight, long before we knew really anything about inhibition. We now know what Gowers didn't know, and that is that there are about 20% of the neurons in the cortex are these inhibitory neurons. We collectively call them the cortical interneurons. These cells provide the primary source of inhibition in the brain by releasing GABA. There's many different types. They have unique morphologies and connectivity patterns. They express different markers, gene expression profiles, and they have very distinct physiologies. They're also lost or become dysfunctional in many forms of epilepsy. And sort of a classic example of this comes from the temporal lobe epilepsy literature. I have here on the right, my pointer doesn't work here, but I have an example from Carolyn Hauser's work in human temporal lobe epilepsy. These are images from the hippocampus, and you can see the arrows pointing to the dentate hilus. And many of the neurons in the hilus are missing in this case of temporal lobe epilepsy. Many of these cells are GABAergic. And so we've been working with this question of can we graft new GABA neurons into the injured brain or damaged brain? And the way we started with this is to dissect out portions of the embryo, the mouse embryo, graft these cells into the mouse brain, and we've put these cells now all over the mouse brain. I'm going to focus on some of our data looking at hippocampus and have used this as a way to create new interneurons in the context of epilepsy. And so the origin of this sort of therapy, I just want to give a brief history of the MGE cell therapy. really came from some foundational studies in the late 90s. Work from John Rubinstein's lab at UCSF showed that interneurons are born about 2/3 of the way through development in this area of the developing forebrain called the MGE, or the medial ganglionic eminence. This was 1997. And then a couple of years later, Arturo Alvarez-Boya's lab showed that if you can take the MGE out of a developing embryo and you can dissociate the cells and implant that into a host animal, and these cells will disperse and migrate long distances in a host brain and survive. And then in the mid-2000s, Scott Baraben's lab from UCSF showed that these MGE grafts produce new functioning inhibitory interneurons when grafted into a newborn mouse neocortex. And these cells will develop and mature and are capable of increasing inhibition in the host animals. And where I got involved really was to develop this as a new therapy for epilepsy. And in 2013, I worked with Scott and Arturo and John, and we developed these GABA cell grafts as a way to stop epilepsy in a preclinical mouse model of drug-resistant epilepsy. And this is really a truly disease-modifying therapy. Over the years, we've shown that this approach is capable of stopping seizures in mice with pre-existing epilepsy. We did this in a pilocarpine model of temporal lobe epilepsy. And we've also shown that this approach can prevent seizures before they emerge. And we've done this in a brain injury model of post-traumatic epilepsy. In both cases, you can see the green cells here are the other grafted interneurons. And you see they migrate all over the injured hippocampus. They integrate quite well, and they appear to survive for the life of the animal. And so our treatment target is spontaneous seizures. I work in epilepsy, and our goal is to develop treatments for epilepsy. So our target is spontaneous seizures. The seizures in these animals, this is just an example from the brain injury model. They are spontaneous. They have an EEG profile. They are convulsive seizures with a behavior that matches the Racine rating scale. They lose their posture. They fall over. On the EEG, the seizure concludes. It goes into a depression. These are clinical seizures in these animals. Most of the animals will develop epilepsy in brain injury. All of the animals will develop in some of the temporal lobe epilepsy models. We don't quite get that same effect in the brain injury models. But when we transplant MGE cells into these animals, we have never seen a seizure in these brain injured animals. And so these animals remain completely seizure-free. In addition to epilepsy, we've looked at a number of different behaviors. We make our transplants into the hippocampus, and so many of these effects that we see are on hippocampal sort of dependent behaviors. So memory is a big one. And we've seen effects in both spatial memory in a number of different tasks. Shown here on the left is a classic Morse water maze where mice with epilepsy have difficulty performing this task well. We can, when we transplant MGE cells into the hippocampus, we can completely correct this behavior. Interestingly, if we transplant MGE cells into the amygdala, there's no effect. And in addition, we've shown effects in contextual memory. This is a fear conditioning task that Farah introduced. Brain injury animals, they have sort of a generalization of their fear memory. Their memory is too strong in a context where they didn't receive an aversive stimuli, and we can correct this with MGE transplants as well. And a number of other behaviors become corrected. And the mechanism for these types of therapeutic effects, at least preclinical effects, seems to be restored inhibition. And so there's an example of patch clamp recordings where we have performed voltage clamp recordings from the dentate gyrus, the granule cells, and after TBI, there's a massive reduction in inhibition. And following MGE transplantation, we see that this inhibition is restored to these granule cells. And we've done this in a number of different brain areas. This is just one example of many that we've done over the years. And in each case, MGE transplantation within a month or two increases inhibition and restores it to control levels. If we look at the grafts themselves, what types of cells are we producing with these transplants? We've looked at many different levels, and at each level, anatomical, electrical, molecularly, these graphs are producing exclusively physiologically mature and functionally integrated GABAergic neurons. The cells express parvalbumin, somatostatin, which you would expect from the medial ganglionic eminence, because this is the area of birth for these types of cells. The cells occupy areas that you would normally find interneurons located in, so for example, the hilus of the dentate gyrus. And then electrically, they display a number of electrical profiles that you would expect from these cell types. For example, fast spiking cells. And we can take the cytosol out of the cell after we record from these cells and get a molecular profile, and these cells express genes that you would expect from a fast spiking cell. So they express parvalbumin, for example. And they don't, this is very important, they don't express genes of other cell types. So other interneuron cell types or glutamatergic neurons or whatnot. And so this is a very specific type of graph that we're performing. At the network level, these cells seem to integrate into the local network, both anatomically and electrically. On the left here, this is work from Scott Barabam. A number of years ago, did electron microscopy and showed that these GFP-labeled cells will form synapses with unlabeled host neurons. These transplanted MGE cells appear to integrate mostly and form context mostly onto host principal neurons. And we can functionally test that either with optogenetics or paired recordings, where we can evoke action potential firing in the transplanted cells and see the voltage response or the IPSC in the postsynaptic glutamatergic neuron. In all cases, these evoked currents and these evoked synaptic sort of events are indistinguishable from what you would expect to see from native host-borne neurons. At a circuit level, we've started to map the connections of these cells. This is the brain injury here. You can see the brain injury there. The transplants were made into that injury. There's the hippocampus in the middle here. And what we're going to do is we're going to scroll through the brain. So all these white cells are the neurons that are connected to our transplanted neurons. This is the perforant path that you're starting to see emerge from the internal cortex. And all these white projections, this is all the input to transplanted neurons. Here's the injured hippocampus here. The red cells, these are the transplanted neurons. The dentate is right here, and you can see a lot of neurons in CA3. So really extensive integration of these cells. We're going to scroll to the contralateral hippocampus in this mouse. You'll see input coming from contralateral CA3. We'll scroll back, and we can trace the axons down the fimbria fornix pathway and see that these cells are receiving input not just from the internal cortex and hippocampus, but this is the medial septum, the diagonal band, and they even receive input all the way out into the brainstem. This is the raphe nucleus, the serotonergic neurons of the raphe. And so really these cells, when we integrate, they're integrating into the entire brain when we put these cells in. We think that this is very key to the therapeutic effects that we're seeing. Okay, so we wanted to test this more directly, and so what we did was we tried silencing these cells and tested what these cells would do. And we did this in a contextual fear conditioning task where animals are placed in a novel environment, receive a foot shock. They have a conditioned response, which is freezing. Like I said before, in these brain-injured animals will have a increase in their fear memory generalization. And this is corrected with the MGE transplants here in pink. But these cells contained the inhibitory dread, and if we gave compound 21 to these animals, what we saw was that the phenotypes returned. So during the task, when these MGE cells are silenced, these animals now are indistinguishable. These transplanted animals are indistinguishable from brain-injured animals. And so this is really strong evidence for the need of the electrical integration of these cells to achieve the therapeutic effects. These effects are not necessarily directly related to stopping the epilepsy, We've tested MGE transplants in a number of other animal models. This is an example of a CHE2 mutant mouse that has a reduction in parvalbumin and somatostatin interneurons, performs very poorly on a number of behavior tasks, the object location memory, object recognition memory, but we've tested many others. Transplants into wild-type animals doesn't really have any effect, but when we graft MGE cells into the hippocampus of these CHE2 mutant mice, we can rescue the hippocampal memory phenotypes in these animals. And so that's great. So that's all mouse cells into mouse brain. And so the question that's emerged is how do we move this forward? How do we can we get similar effects with human pluripotent stem cells, which might be a little bit more of a translational cell source? For obvious reasons, they can be made directly from patients, avoid some of the ethical concerns of fetal tissue. And the approach that most people will take is this one, where we try to recreate with the morphogen gradients that are naturally found in the developing brain. So, for example, in the dorsal forebrain, the pallium, Wnt and BMP signaling is high, and this promotes the generation of pyramidal neurons. In contrast, in the ventral forebrain, sonic hedgehog is high, and that signaling promotes the generation of the MGE and the inhibitory GABAergic neurons. And so high sonic hedgehog and low Wnt BMP signaling is the approach that most labs have taken to date. And there are many protocols that have been developed for deriving human MGE-like cells. I split these sort of into first generation and second generation. The first generation came out about 10 years ago, and the second generation has started to appear in the last couple of years. And more or less, a lot of these claims are quite similar although there are some distinct differences between the different protocols, but claims of making a pure or nearly pure population of GABAergic neurons, high percentages of somatostatin interneurons, and low generation of parvalbumin cells. Some of the challenges that have emerged is that interneurons grown in vitro are very different from interneurons that appear in vivo. And this is really highlighted by a group at UCLA that took and examined a number of these in vitro protocols for generating human interneurons and compared them to the fetal human brain and interneurons in the adult. And you can see some of the RNA sequencing here. In the comparison, there's almost no overlap here between the protocols generating interneurons in vitro and what you see in vivo. And they concluded, sort of at the end of the paper, that none of these methods produce mature postnatal-like inhibitory interneurons. And some of the challenges that show up are things like this, partial cell fate specification. And what I mean by that is we can grow neurons in a dish, and they can express many of the genes that we would like to see, but they often express genes that we don't want to see. So, for example, PAX6 expression in a population of interneurons would be not appropriate, given that PAX6 is almost exclusively expressed in excitatory cells. Or incomplete cell characterization. And human neurons take a long time to develop, either in vitro or after transplantation. And this has led to a little bit of ambiguity in sort of characterizing their cell properties and obtaining appropriate electrical profiles. And so I agree with my colleague here, Joy Sebi, who said, I do think there should be no doubt if you're going to put this into people's brains. And the reason I say this is because the type of interneuron really matters. And this is a great example of this. There are many different types of interneurons, and MGE transplants are therapeutic, but there's other types of interneurons that come from the caudal ganglionic eminence, and these cells are not therapeutic. This area of the embryo produces reelin and VIP interneurons, not parvalbumin and somatostatin interneurons. We can transplant these cells as well, and these cells will migrate and disperse and integrate as interneurons, but generate these VIP interneurons, a large population of them. And these cells target other interneurons. They don't target principal cells. And if we transplant these cells into wild-type mice, within a month or two, 3, the animals start to have spontaneous seizures. And so this I see as sort of a cautionary tale for why the type of interneuron really, really matters. And so given some of these challenges, one of the obvious questions that has come up, at least for us, is are stem cells even needed in the first place? There's a number of non-human fetal cells that exist. Of these, pig is probably the most promising candidate. And this is simply because there's a lot of progress in the field in generating humanized pig cells and pig organs that might be used for transplantation purposes. And if you compare the developing embryo of the mouse, the pig, and the human, you see a lot of similarities. The MGE is born in the same place. The cells are born in the same place, express a lot of the same transcription factors. Scott Barabin's lab has come up with methods to dissect out the pig MGE and transplant these cells into rodent models. This is a rat, and these green cells here are the pig MGE cells. These cells disperse all over the hippocampus, just like the rodent cells, express genes that you would expect from MGE, for example, somatostatin. They migrate quite a ways, but are they therapeutic? And so I want to end with a case study, but a case study from a sea lion. This is Cronut. Cronut is now a very famous sea lion. In 2017, he was first beached in San Luis Obispo County, and he was rescued a number of times by the Marine Mammal Center. He was found disorientated and confused. He was found on the beach. He entered a couple of times into the public restrooms. And the diagnosis was a suspected domoic acid toxicosis due to the algae blooms that happen on the coast of California. At a given point, they rescued him a number of times. They could no longer release Cronut into the wild, and they couldn't keep him at the Marine Mammal Center, so he was transferred to Six Flags Discovery Kingdom in Vallejo, California, and he started to have seizures. MRI showed atrophy of the left hippocampus, and over time, his condition started to worsen. He stopped eating. As a result, he was losing weight and started to have multiple seizures. He was started on a number of anti-seizure medications, but in 2020, at the height of the COVID pandemic, his condition had really started to fail, and they were considering euthanasia. And at this point, the Marine Mammal Center called Scott Barabin to ask about a MGE transplant to save Cronut's life. And I will never forget this because September, my phone rang, Scott was on the other end, and he didn't even say hello. He started off with, "You got to talk me out of this." Yeah. I didn't. And in October of 2020, a group of about 20 neurosurgeons, vet staff, scientists came together. You can see Cronut here. He became the first patient in the world to receive an MGE transplant. And here Cronut is getting being prepped for surgery. Here he is after the surgery. And today, he's made an amazing recovery. And so this is, it's now 2023. He's over 3 years seizure-free. He's eating, as you can see. He's following directions. He's able to do tricks. And before surgery, he was never able to do any of these things. Keep in mind, they don't designate sea lions for euthanasia without a lot of consideration. And so this is sort of an amazing sort of proof of principle in a large animal for the therapeutic potential of this approach to treat epilepsy. Yeah. And so where are we now? In my view, I see MGE transplantation as a really promising disease-modifying therapy for epilepsy. MGE cells migrate and fill entire brain areas. They survive for the life of the animal. We're wiring these cells into the entire brain. I think that is a foundational sort of mechanism for how we see some of these therapeutic effects. But stem cells simply have not met the level of rigor that we've seen with the fetal MGE progenitors, mainly in rodent and pig. And so I think as we think about the future, I would urge caution before racing to the clinic with stem cell-based approaches, simply because there are a number of challenges that are emerging in the stem cell field in terms of cell purity and efficacy, and it's simply unclear if all of the sort of characteristics of these cells really, truly, faithfully match what we see with fetal MGE progenitors. So I'm out of time, so thank you very much.
That
was a fantastic presentation. Thank you, Dr. Hunt. And now it is my pleasure to invite Dr. Vikram Rao, MD, PhD, who is an Associate Professor of Clinical Neurology, Ernest Gallo Distinguished Professor and Chief of the Epilepsy Division in the Department of Neurology at UCSF. Dr. Rao's clinical and research interests involve applications of neurostimulation devices for drug-resistant epilepsy, neuropsychiatric disorders, and seizure forecasting. Welcome.
Hi, good morning, everyone. I can't believe I have to follow Krona the sea lion. That is tough. So I want to thank Drs. Lowenstein and Patel for including me in this great symposium. Our task today is to review the current state of neurostimulation for epilepsy, and then we're going to try and provide a glimpse of what the future may hold. These are my disclosures. They include current and previous work with NeuroPace, LivaNova, Medtronic, and Enlighten AI. These are our learning objectives. We're going to start by highlighting some of the current challenges and limitations of using neurostimulation for drug-resistant epilepsy. We'll look at how characteristics of brain networks can shape the effectiveness of these therapies, and then we'll talk about barriers to equitable access to these therapies. Dr. Knupp showed you this famous slide earlier today, and I'm going to show it to you again because I think it makes a very important point that historically we as a field have been very good at developing new anti-seizure medications. But I would argue that we've been probably less good at moving the needle on rates of drug-resistant or medically refractory epilepsy, rates that have stayed disappointingly constant for 150 years. Fortunately, though, over that time, we have made considerable progress in our ability to localize focal seizures using all of the techniques shown here, progress also in our ability to resect or to ablate the seizure-producing brain tissue, often with excellent clinical outcomes. We now have randomized controlled trial data in both adults and children demonstrating that epilepsy surgery can be superior to medical therapy. And so we like epilepsy surgery. We offer it really whenever we can. The problem is that there are times when we can't. And those limits of epilepsy surgery are evidenced probably most famously by this gentleman. This is HM. You know him by his initials if you don't know him by face. HM had epilepsy, probably post-traumatic, and he famously underwent a bitemporal resection, which actually improved his seizure control, though he wasn't seizure-free. But of course, it left him with a devastating anterograde amnesia. And his case really highlights the fact that epilepsy surgery, though good when it's possible, is not always feasible. In scenarios like that, we are now increasingly turning to use of one of several implanted approved neurostimulation devices. In the US, there are 3 approved neurostimulation devices: vagus nerve stimulation, or VNS, thalamic deep brain stimulation, or DBS, and responsive neurostimulation, or RNS. 3 very different devices that have in common the fact that they deliver electrical energy to the brain to reduce seizures over time. Now, the mode by which they deliver electrical energy to the brain can be broadly categorized as being either open loop or scheduled intermittent stimulation, or closed loop, where stimulation is delivered in response to detection of abnormal activity of some kind. Now, unlike epilepsy surgery, seizure freedom is uncommon with any of these devices, but we do see very encouraging reductions in median seizure frequency in clinical trial cohorts. The problem with these median seizure frequency reductions, though, is that it really belies the underlying variability in clinical outcomes across individuals. And I've tried to show that here. So each bar is a patient. These are drawn from different studies. But in red now, I've highlighted the non-responding patients, the patients who did not even have a halving of their baseline seizure frequency. And in green, the responders, the people who had a 50% or greater reduction in seizure frequency. And the purple arrows really highlight this striking outcome variability across individuals. And this really presents a dilemma for the clinician. How are you supposed to select the right neurostimulation device, the one that has the best chance of reducing a patient's seizures? And even once you choose a device, how do you know for a given person if they're more likely to be on the left side of these plots or on the right side of these plots? Well, the answer is that it's not that easy. Let's say you were considering RNS for a given person, and you wanted to know, is this person likely to become a super responder with a 90+% reduction in seizure frequency? Or are they going to be a non-responder or maybe some intermediate response? How can I counsel them? Well, unfortunately, there really aren't any first-order clinical variables, demographic features that will help you stratify these outcome possibilities, things like onset age of epilepsy, how long you've had epilepsy, duration of stimulation or amount of stimulation, where you stimulate. Really, none of these things reliably stratify clinical outcomes and help you prognosticate. Now, in scenarios where the first device that's chosen doesn't yield the desired effect, patients are increasingly turning to treatment with a second neurostimulation device and rarely even a third implanted neurostimulation device. And all of these reports are just from the last few years. And I suppose, on the one hand, it teaches us that failure of one neurostimulation device does not necessarily portend failure of all of them, which is good. But I would say it would be even better if we were somehow able to rationally choose the device that was optimal for a given person on the front end, and thereby spare them unnecessary procedures, unnecessary morbidity. The problem is that is easier said than done. I think any medical therapy is really only as good as our ability to identify the patients who are most likely to benefit from it. But device selection is challenging. There have been valiant efforts to make device selection more algorithmic for clinicians, and I applaud these efforts. They are really helpful. But the sheer number of arrows on a diagram like this really underscores what a tremendous clinical challenge this is. This is really our current state of affairs. We don't have a rational means, a data-driven means of prognosticating outcomes, how to select the right device for the right person, and therefore we don't have a great way to optimize outcome. In my mind, this dilemma really mandates a fundamental reappraisal of the mechanism by which devices work to reduce seizures. Perhaps if we understood mechanistically how these devices work, we'd understand more about how to choose the right one for a given person. Now, it feels almost passé at this point to say that epilepsy is a network disorder because, of course it is. And yet, as recently as 10 or 15 years ago, there were fewer than 100 mentions in the literature to terms that we take for granted now. Terms like epileptic network, seizure network. And yet, our therapies, in one way or the other, do target brain networks, don't they? Resection of the seizure onset zone, for example, might normalize network function elsewhere in the brain. And similarly, neurostimulation, delivering electrical energy either directly or indirectly to the seizure focus, can help mitigate network dysfunction elsewhere in the brain. In fact, in one rubric, you could think about all of our epilepsy therapies along the lines of their site or sites of action within brain networks. So, for example, on the left, laser ablation could be thought of as a therapy that targets nodes within networks, whereas a disconnection procedure like corpus callosotomy is a therapy that targets edges or the connections between nodes. Whereas still other therapies, like all the many anti-seizure medications we just saw, well, they bathe the whole brain in medication, and so they're treating the network as a whole. Now, I've highlighted RNS on this slide on the left here. We'll come back to that. Canonically, we think of that as a therapy that works on nodes. We place leads intracranially at the seizure focus or foci. But this framework really raises an intriguing possibility. Could it be that intrinsic features of brain networks render that network more or less susceptible to therapy with a particular neurostimulation device? Could it be that if we knew how to map network characteristics to a device, that we could more rationally choose a device that will be best for a given person? We're going to explore this idea by considering each of the 3 approved neurostimulation devices. We're going to start with this one, responsive neurostimulation, or RNS. This is the canonical closed-loop neurostimulation device. And the sensing half of that closed loop involves the ability of the device to store chronic EEG. These are long-term intracranial recordings of neural activity of many different types that are recorded in ambulatory patients who are in their home environment, on medication, living their lives over months to years. Really, in some ways, an unprecedented treasure trove of information. The treatment arm of that closed loop was originally conceived as a seizure stopper, akin to an AICD for the brain. The idea was that detection of abnormal activity would trigger delivery of brief, high-frequency pulses of electrical stimulation to terminate incipient seizures acutely. So this is a nice idea, but there are actually several lines of observations now that suggest this may not be the only mechanism by which RNS works to reduce seizures, and I think it's worth reviewing some of these observations. Observation number 1. No AICD should take years to treat an arrhythmia, and yet seizure reduction with RNS takes time. These are data from the long-term treatment trial of RNS showing that by year 9, we see a 75% median reduction in seizure frequency, but you notice the improvement accrues gradually year over year. Even in a subsequent real-world outcome study, we saw again that we're getting good outcomes, maybe a little earlier, maybe a little better, but yet outcomes improve gradually over time. Observation number 2, stimulations with RNS far outnumber seizures. These are data from one of my patients who is treated with RNS. She receives about 1,200 brief stimulation therapies per day, a pretty typical number, and yet her average frequency of electrographic seizures is only 1.3 per month. So it could be that all 1,200 of those stimulations are acutely nipping seizures in the bud, or, more likely, the vast majority of these stimulations are delivered in the interictal state, which is not probably how you would design a seizure stopper. Observation number 3, you would also design a seizure stopper to work best, or maybe only to work, if you're operating directly at the onset of the seizures. And yet we know that, at least in the case of mesial temporal lobe epilepsy, that if you place RNS electrodes either unintentionally or intentionally outside of the hippocampus, outside of the seizure focus, we can still see appreciable reduction in seizures. Similarly, we might think that a device like RNS would work best for the most discrete, most well-localized seizure foci, and that may be true, but we know also that more spatially extensive, less well-demarcated, so-called regional neocortical seizure onset zones can also respond to RNS therapy, and in small series, the outcomes actually mirror those seen in larger clinical trials. And finally, we are learning that there are some atypical stimulation paradigms that can be effective with RNS. Long burst duration, low frequency stimulation of the hippocampus can help people who did not respond to conventional paradigms of short duration, high frequency stimulation, and even open loop or continuous high frequency stimulation of the hippocampus can help reduce seizures in some people. So, where does that leave us? It leaves us where we're often left in neurology, which is that the situation may be more complicated than what we originally imagined. But that is okay, because in recent years there have been a number of studies that are starting to shed some mechanistic light on how RNS and our other devices are actually working to reduce seizures. Let's talk about some of those studies. We'll start with this one. This is work from Brian Litt's group at University of Pennsylvania. They looked at intracranial EEG recordings of seizures in people who went on to get treatment with RNS. And most of those people became responders, some became non-responders. But this enabled them to look at electrographic features of seizures in relation to clinical outcome. And what they found is that a network measure called ictal synchronizability, which reflects the ease by which a seizure can engage a functionally connected brain network, was lower in responders to RNS compared with non-responders. Using a similar study design but a non-invasive technique, magnetoencephalography or MEG, Jolene Fan at UCSF looked at global functional connectivity in patients who went on to be treated with RNS. And what Jolene found is that, as measured by MEG, global functional connectivity in higher frequency bands was actually higher in responders compared with non-responders. So if we take this and the previous study together, it seems that the networks that are most amenable to treatment with RNS are those with high global functional connectivity, but those for which the seizures they give rise to have low synchronizability. This is work from Chris Butson and John Ralston. They asked a very straightforward question, which is, when we do hippocampal RNS, does the connectivity of the volume of tissue you're activating matter at all in terms of outcome? And what they found is that it certainly does. So when the connectivity of the volume of tissue activated engages with areas like the medial prefrontal cortex, the cingulate gyrus, the precuneus, that outcomes with RNS were better. This is work from Dilip Nair's group at Cleveland Clinic. They used stereo EEG in patients who went on to get RNS, and they measured cortico-evoked potentials to define receiver and projection nodes, areas with high inward or outward connectivity, respectively. And what they found is that when you place RNS electrodes near receiver nodes, that outcomes with RNS were better than when you didn't. But these studies somewhat neglect the temporal dimension of network connectivity, don't they? They sort of presume that networks have a static wiring pattern that we can leverage. But we are increasingly becoming aware that epilepsy is a cyclical disorder, that brain networks fluctuate through periods of relatively high and relatively low seizure risk. And that raises the possibility, could it be that the effects of neurostimulation when the network is in one state might be different than when the network is in a different state? And we have evidence for exactly that now. So this is work from Sharon Chang at UCSF. Sharon Chang did a retrospective analysis and statistical modeling of states of high and low seizure risk. And what she found is that when you make a stimulation parameter adjustment on RNS, something like increasing stimulation frequency, if you do that when somebody is in a state of low seizure risk, that's favorable. That tends to keep that person in a state of low risk. But if you make the same stimulation parameter adjustment when the person is in a state of relatively high seizure risk, that's actually counterproductive. It tends to keep them in a state of high risk. More recently, Anderson and colleagues, again from John Ralston's lab, have shown us that when RNS stimulations are preferentially delivered during states of low seizure risk, outcomes with RNS are better. And so these studies together suggest that maybe a next-generation device would do well to be sensitive to underlying network state and deliver stimulation adaptively as a result. That ability to deliver stimulation adaptively is really critical because brain networks are not static. This is work from Ankit Kambadi at UCSF, and Ankit asked a simple question. He said, what's different about people who are poor responders to RNS versus people who are super responders? Because we see both. He said, surely something must be different about their neural recordings over the course of RNS therapy, and in fact, there is something different. The responders to RNS are the ones whose brain networks are more malleable. He saw greater reorganization of functional connectivity in an early and sustained way in people who were responders to RNS therapy compared to those who were non-responders. They had more immovable brain networks. This is direct evidence for a role for long-term neuroplasticity in the mechanism of RNS. But what about our other devices? What about vagus nerve stimulation, deep brain stimulation? Are we seeing similar themes emerging with the mechanism of these devices? Well, in fact, we are. We now have evidence for network biomarkers of response to VNS. That evidence comes from studies of fMRI, studies that use MEG. These studies reveal the existence of brain networks that are preferentially expressed in responders to VNS therapy compared with non-responders. Similarly, with DBS of the anterior nucleus of the thalamus, the ANT, we're seeing with studies of fMRI that responders and non-responders have very different patterns. Responders tend to have widespread cortical deactivation and relative activation in limbic networks, default mode networks, compared with non-responders. Aaron Warren and colleagues looked at seizure reduction with centromedian thalamic stimulation in a setting of Lennox-Gastaut syndrome. And they found that the connectivity of the volume of tissue you activate in the thalamus matters a lot. When that connectivity engages areas shown in yellow and orange, in these maps, seizure reduction was greater than when it engaged blue and purple areas. And this enabled them to identify a so-called sweet spot for stimulation of the centromedian thalamus based on the connectivity of that area. Using scalp EEG and concurrent ANT DBS, Aiello and colleagues recently found that theta range connectivity, thalamocortical connectivity, was a biomarker of response to DBS. It predicted good outcomes. And finally, this is work from Mike Fox's group. They have recently revealed the existence of a putative brain network for lesional epilepsy, a network of structures wherein lesions were more likely to result in epilepsy compared to lesions outside of this network. And remarkably, what they found is that in patients who were treated with ANT DBS, the volume of tissue activated, when it had connectivity to this lesional epilepsy network, that the efficacy of DBS was greater than when it did not have connectivity with that network. So we have covered a lot of ground. Maybe we can start to integrate some of these findings and build a model for what might be going on. Well, I don't claim to have all the answers, but I do wonder if perhaps some of the reason that we see such variability across individuals with all of our devices might relate, at least partly, to the fact that we currently select and manage these devices largely agnostic of patient-specific network characteristics. Characteristics like the connectivity of the network, key structural and functional nodes, the momentary state of the network, and the capability of the network to undergo long-term reorganization. Perhaps if we were more mindful of these network features, we would be able to tailor our neurostimulation therapies accordingly. If we revisit this slide on the site of action of epilepsy therapies, we had put RNS as a seizure-stopping therapy that works on nodes within networks. But perhaps you'll agree that this might be more properly classified as a therapy that reduces seizures by virtue of its effects on brain networks. And as we turn an eye then to the future, where are we going with all of this? I can imagine that we will increasingly be using network assays that involve EEG or MEG or fMRI or other techniques on the front end to characterize patient-specific network features, to leverage that information to select the device that is most likely to help a given person. and therefore spare them empiric trials of multiple devices. This, in my view, is the dream of personalized therapy in neurostimulation, and I think this is the way that we will ultimately optimize outcomes. But even as we work on optimizing outcomes, we have to be mindful to also optimize access to these therapies. Access that is shaped by many factors: insurance, geography, socioeconomic status, racial and political factors. We know all of these things play a role. We know that, for example, most patients access neurostimulation therapies through academic medical centers, often in wealthy metropolitan areas. And we know that, at least in the US, average household incomes in counties where stimulators are implanted is higher than average household income in counties where stimulators are not implanted. We know, for example, that things like spinal cord stimulators are used more often in patients with private insurance. We know that there are significant racial disparities in utilization of DBS. We know that equitable access to neurotechnology is a multidimensional challenge, and I certainly don't claim to be an expert on all of these things. There are many people at this meeting who have far greater expertise than me, but we have to think about the underlying ethical principles of equity, resource allocation, distributive justice, and work as a community of leaders in neurology and epilepsy to try and ensure that we are optimizing access even as we optimize the therapies themselves. And so, to summarize the impact on clinical care, we looked at how network features can predict outcomes with neurostimulation. We talked about the prospect of leveraging brain network features on a per-patient basis to personalize neurostimulation device selection and therapy management. And then we highlighted the importance of ensuring equitable access to these therapies, which remains a critical unmet need. And so I will stop there. You're going to hear from Dr. Lowenstein next, who's going to bring it all home, so you're in for a treat. Thank you.
I'm just going to move this over. Yeah, thanks. That happens a lot, doesn't it? Yeah. Thank you, Vikram. That was a great talk, as have all the talks been. And, well, now, as you heard at the beginning, Manisha gave me the unenviable task of trying to imagine the future care of the patient that Kelly described at the start of the symposium. And I just decided to take the assignment actually rather literally, and that is to imagine what the care of Kelly's patient might look like decades from now. So these are my disclosures. And I want to highlight the fact that just as a core theme of this presentation is the AI revolution, I relied very heavily on the AI language model ChatGPT-4 and the AI image generator DALL-E in pulling together much of what you're going to see. So, what about imagining the care of our patients, say, 27 years from now, in 2050? Now, no doubt most of you have heard the quote that it's a fool's errand to try and predict the future. And here are just two examples of that truism. First of all, this is Thomas Watson, who is the chairman and CEO of IBM, and you'll see that in 1943, he said that I think there's a world market for maybe 5 computers. But especially like this one, in 1962, when Decca Records rejected the Beatles, claiming that, quote, guitar groups are on the way out, and that the Beatles had no future in show business, this proved to be one of the most infamous misjudgments in music history. But I'll also have to say that in the interest of full transparency, I need to admit my colossal failures in this regard as well. Manisha somehow thinks I'm a futurist. I'm not. One example, the Segway in 2001. I was just certain that any urban image would show everybody tooling along on these things. Definitely didn't happen. You can take a very nice tour of Golden Gate Park on Segways. And I actually saw a Segway here at the Convention Center, I think, yesterday. And then the other one was in 2012, Google Glass. I was also sure, looking at an audience like this, that we'd all be wearing these things, and boy, that was wrong. But I have had at least one success, I think, and that was the prediction of the iPhone. Introduced amazingly, if you think about it, in 2006, that's only 17 years ago, which placed on the foundation of the internet with its instantaneous and ubiquitous access to information and worldwide connectivity would perhaps be as transformative as a Gutenberg printing press? Well, history will determine that, but I do think it's amazing to think. Imagine if an alien anthropologist had come and visited and studied Homo sapiens, say, in the latter part of the 20th century, and then came back for a comparative study, say, now. I mean, it would probably document the fact that Homo sapiens have evolved this rectangular appendage that typically sits on the end of their right hand. If sitting there, people are commonly looking down at this device, and if walking along, they're holding it to their ear, whatever. With the developments over the past year, and this is a Dali image, actually, of what the world might look like. With the developments over the past year, I'm convinced, like many others, and you've heard allusions to this already, that we are at the dawn of what may be the biggest inflection point in societal evolution since the Industrial Revolution, and that is the advent of true artificial intelligence. So, in thinking about the way that AI and other technologies will weave their way into the world of patient care, here are the main elements of the storyline. First, there will be comprehensive AI-powered initial medical analysis when a patient seeks help. This will then lead to pre-visit diagnostic tests. There will then be a virtual reality epileptologist consultation with real-time AI documentation and analysis. And this will culminate in personalized data-driven diagnosis and treatment recommendations. So, here's our patient. You heard about her from Kelly. She's now a 29-year-old woman referred by her primary care health professional who's in Silverton, Colorado, in a rural area, for a recent unprovoked generalized tonic-clonic seizure. Now, she has a distant history of febrile seizures and an unprovoked GTC when she was 18 months old, and the evaluation back then, in 2023, showed no abnormalities other than 2 variants of unknown significance, and she has no other known medical problems. So first off, in the care of this patient, the comprehensive AI-powered initial medical analysis. Here, a sophisticated AI system provides an intricate analysis of the patient's lifelong medical and social information, deciphering all medical evaluations to the present, all laboratory studies, including imaging, and was now routine genomic, transcriptomic, and proteomic profiles, just to name a few. With this information, there are pre-visit diagnostic tests, and based on the updated AI analysis, the patient is asked for her permission to undergo new studies needed for evaluation, including a state-of-the-art brain imaging epilepsy protocol, which of course is just beyond what's already been happening to everyone, which is whole body imaging, which is standard. But she'll also have a microbiome analysis and an assay for circulating biomarkers with high predictive values for epilepsy susceptibility, diagnosis, and prognosis. In addition, the patient is provided what has now become a standard cerebral monitoring device, which is a consequence of major breakthroughs in physics and materials science. And this utilizes implanted subscalp, self-powered microelectrodes, enabling at least a week or more of continuous monitoring prior to the actual clinic visit. And finally, the patient undergoes a home-based neuroexam directed by an AI assistant, which provides instructions and files high-fidelity recordings of the basic exam, including things like pupillary responses, eye movements, facial symmetry, fast finger movements, and gait. And importantly, all of these assessments have been pre-authorized because this patient, like all patients here in the U.S., now have excellent quality health care through the nation's single-payer system adopted back in the mid-2030s. And I realize that what I just said may be the most preposterous, if not delirious, aspect of my presentation, but whatever. We got a dream. And equally important, high-speed internet is universally available, which in combination with universal healthcare coverage has removed virtually all the disparities in access to care, and that's not just here in the United States. So, next comes the virtual reality epileptologist consultation. So, distance from expertise is no longer an issue. In this virtual space, both the epileptologist and the patient are represented by hyper-realistic avatars. The doctor uses a virtual display to showcase various results, including a summary of the relevant history, the neuroexam, and the lab findings. And in the background, the AI assistant is recording the entire visit, enabling the patient and the doctor to actually spend the entire time focused on one another, rather than worrying about documentation and other distractions. They'll be able to talk about the questions that Kelly brought up in her interactions with her patients, the questions that patients and their families desperately need time to try to understand and listen to. And the image, this image, of the doctor with his back to the patient, focusing on entering information on the computer screen, which really characterizes healthcare in the early part of the 21st century, has finally become a vestige of the past. And finally, there's the personalized, data-driven diagnosis and treatment recommendations. So nearing the middle of the visit, the patient and the doctor turn to the AI assistant for an interim summary and draft assessment, which incorporates all the information on the patient accumulated to date, along with a population-based analysis of millions of people looking for individuals who may have a history and phenotypic and omic profile similar to this patient. And in fact, it becomes clear that a combination of 4 and possibly 5 rare genomic variants, including one of those variants of unknown significance that was identified back in 2023, along with all the testing to date, verify the diagnosis of a genetic epilepsy. And the doctor and the AI assistant next provide a careful description of the basis for the diagnosis and the rationale for therapy, and they take into account the patient's educational and cultural background. And then there's a discussion of preventive measures such as sleep hygiene, stress reduction, and so forth. And importantly, the treatment plan. Based on the omic profiling and the other lab data, along with the response of other patients with the same genetic cause, the recommendation is the manufacturing of a personalized antisense oligo cocktail. God knows what this will really be in 2050, but there will be something. And this will become available within 3 to 4 weeks. It takes time to manufacture. In the meantime, the patient will take a 5th-generation anti-seizure drug that also fits the patient's profile, including the microbiome and pharmacogenomic data already in hand, and it also happens to include a nanochip that reports drug absorption. And again, these things take a bit of time, but no problem in getting hold of that medicine because it's delivered the same day by drone. And finally, standard follow-up care of this patient will include the continuous monitoring of cerebral function with the device we talked about, analysis of sleep behavior, simple neuroassessments, and regular check-ins with the AI assistant. So there you have it. Here are my main points. The AI revolution is happening, and will begin to have a very real impact on the nature of epilepsy care soon. In some ways, it already has. The integration of large data sets, for example, from social context to clinical history to laboratory results to population data, combined with AI assistance, will enable us to provide far more individualized care. Virtual connectivity and AI input will reduce the access gap that is part of the current disparities in healthcare, and the, quote, in-person interaction between the patient and their healthcare professional will finally return to what all of our patients deserve, and that's an informed, caring, compassionate, meaningful connection. And finally, ongoing advances in science, no question, are paving the way for incredibly effective treatments and cures. And with that, my AI assistants and I want to thank you. So I'd like to, Manish and I would like to invite our speakers up, and we'll now have some time for Q&A.
That was fantastic.
Thanks.
So if you have questions for us, please come. There are some microphones here that could be distributed, or please come up and ask questions.
That was fun. Thanks.
And our entire panel is really pleased to address any questions. Please go ahead there.
Okay.
Green shirt.
Hi, Dylan Verdon from KIF1A.org. We're very fortunate because we are looking at ASOs and gene therapies, and even have some that we are using in a child with our disorder, and it's showing really promising results. When we presented the prospect of ASOs to the rest of our family community and started talking about intrathecal injections every 2 weeks for the rest of your life, looking at the parents and their families, they really had to think about the logistics and also really the burden of treatment. And so as we think about expanding these therapies and getting these therapies more precise and better suited towards particular diseases, how do you think about the role of delivery and its role in equity, and what prospects are there for making those treatments more easily administered and more accessible to people so that it's not so invasive and not such a life-changing burden in and of itself?
Katie, do you want to go first?
I'll take, I guess.
And then I'm happy to.
Oh, yeah, you use your mic.
Yeah, I think I have. Is my mic on?
Yeah.
Can you guys hear me? No. Okay.
No?
No.
Mike?
Mike.
Is it?
Try again.
All right. Can you hear me now?
Nope.
Just use this.
Nope.
Yeah,
All right.
now we can.
Where's your mic on?
You can hear. Yeah. I'm not sure which mic we're using.
Oh, now you're on it.
I mean, you know, what we're seeing in trials now is that families are pretty open to intrathecal injections if there's some hope of benefit for their children. Every 2 weeks is a lot, but there are devices that can deliver this as well, so that, you know, and so my hope would be if something has to be delivered every 2 weeks, that you would think about using a device that can do this, similar to like a baclofen pump or something like that, so that families can come in, get dosed, and go home for extended periods of time. Every 2 weeks is quite a bit, and being able to access health care at an institution that can do that could be quite challenging for some families, particularly in Silverton.
The middle.
Bill.
Oh.
Hi, Bill
Okay,
Theodore.
Bill.
So Vikram, your talk was great, but I've always been a bit troubled by the claims made by devices of continuing improvement over time. And perhaps you could say a little bit more about the controls for those long-term extension trials, the issue of dropouts, loss of patients to contact, and how solid those reports of an increased response really are.
Yeah, thanks for this. Let's see. Okay, there we go. Thanks for that question. I think it's a really good point that you're making, which is that you have to be quite mindful of interpreting data that are open label long-term extensions that are plagued by attrition and other factors that can make it seem like outcomes are improving over time. Even after you control for those things, you do see that most of our neurostimulation therapies do actually improve over time in terms of their efficacy. We see it also on a per-patient basis, sometimes seeming to do nothing and leaving device settings constant. You actually see gradual reduction in seizure frequency as well. But I think you're very right to have a healthy dose of skepticism to interpret data that are outside of the kind of controlled circumstances of the initial trial. But even so, I think that there is something there, and we see it quite consistently across devices.
Hi, this is a question for Dr. Hunt. I, like many people in the audience, were touched by your story by Cronut. Could you tell us a little bit what species of cells you used and how long it took for you guys to observe effects in the seizure decrease?
Okay, am I on? Okay. So the species of cells were pig. They were pig embryos, fetal cells. In terms of seizure monitoring in a sea lion, it's not straightforward. So it's not like a rodent model where you can start recording EEG right away. And so this is just periodic sort of monitoring. There's a case study, if you're interested in this, that's been published that goes through a lot of those details. But after a few months, the cells integrate, they mature, and you start seeing therapeutic effects. Yeah, he started eating sort of right away, though.
at the end there.
My question, great talks, everyone. I work in an area with underserved patients. We have somewhat limited resources. Vikram, what workup would you suggest before device placement and device selection in a place where we have somewhat limited resources? Like MEG isn't really an option for us. What would you recommend?
Gosh, it's a hard question. It depends so much on the characteristics of the individual, but one thing that you could say is that some devices require you to know exactly where the seizures come from, or at least we think that they do, like RNS. Other devices, that's not necessarily obligate, so the workup that you have to do to use VNS or even thalamic DBS, you could argue is less because seizure localization is not obligate with those devices. So perhaps if it was a scenario where you weren't able, for logistical reasons, to do the full workup that you'd like to do to pinpoint seizures, that might sway you towards a device where seizure localization was not necessary.
Middle.
Hi, Dr. Friedman from New York Medical College. I have two questions. One is on the RNS stimulation. So I thought that was really interesting how you were talking about their different effects depending on the energy state. So the question comes up about women and sex differences in their hormone cycle. Is there an optimal time to deliver the stimulation during estrus or other parts of the cycle that influences their seizures? And then the second question is involving the antisense oligonucleotides, which I remember working on that many years ago in some of my earlier studies. And so when you knock down an RNA, so the effect, first of all, it's very expensive, so how it can be sustained clinically. But also, does anyone know what the really long-term effects are by knockdown in terms of compensatory mechanisms like you would get with a mutant? So, yeah.
After you, Vikram first.
The question was about sort of state-dependent effects in relation to hormonal cycles, and I think it's a very good question. The short answer is we don't know, but I think your idea is a very good one. We know that hormones can influence cortical excitability. We know that hormones fluctuate, and we have evidence that neurostimulation effects are state-dependent, or can be. So it's a very reasonable idea that there might be certain times that are more or less opportune to intervene with stimulation.
Yeah, and to address am I on? No. Nope. How about now? No. How about now? I could shout really loud.
There.
Yeah, now we're good? Great. And to address the ASO question and knocking down a particular gene or protein and how long is that sustained? So I think this ties to the repeat dosing question. I think that one of the issues with ASOs is that they do wear off over time. And so there will need to be repeat dosing. I agree with Kelly. I think in the future that might move to devices. But to address your bigger question of what are the compensatory effects of that, honestly, we just have no idea. Right now, there are 2 or 3 studies in preclinical models, and I think we just don't know. I think we need to dose at different times. I think we need to look at continuing to look at ECOG and how do the seizures change, but then I also think we need to be looking molecularly at how the protein abundance is changing. And dispersion as well. I think that's another big question, getting to the network question. So lots of answers to say, I don't know.
Question at the end.
Hi, my name is Verena Borwinkle, and I'm from the University of North Carolina. And I really want to applaud the planners of the committee to incorporate disparities and technology to overcome these. And I might add that in my own experience in helping set up a stroke network in Latin America, that I think a lot of the technologies we're talking about are already here, and it's incumbent on us to be open-minded to incorporate them. For example, in the United States, we already have a resting state functional MRI hub and spoke network for epilepsy out of the University of North Carolina, where, for example, any patient in the United States could receive this seizure localizing benefit through local MRI acquisition and hub and spoke expert interpretation at other centers. So I would just love to hear from the panelists how at your centers you're already incorporating such technologies and where you see us now and in the next year.
Maybe that's me.
I can start. So
I'm on now, okay. So obviously through the pandemic, we've been using telehealth, which means that a patient in Silverton, as long as they can find internet to access, because it can be a little bit tricky, we can do telehealth visits. We also do, we will have the MRI that's done locally sent to us. We also read EEGs remotely, so patients don't actually have to travel for those types of things. But for more detailed evaluation, unfortunately, those patients do have to travel to us for admission to the epilepsy monitoring unit and such. But I think there's, as insurance companies are allowing us to do more and more remote care, I think we're going to see more and more access, in the upcoming months, years. So I think that'll be an exciting change. I don't know what everybody else's experience has been.
Yeah. And I maybe.
You might.
Mike. Yeah. Oh, yeah, great. I can probably just add from the genetics genomics perspective, in terms of access to genetic testing, it's relatively straightforward to ship somebody a genetic test, have them sent back to do telehealth in terms of delivery of results. I think the challenge that we have is in genetics education, and I think that is very true in the United States and very, very true across the rest of the world. And so I think where we really need to be investing our efforts is in genetic education, getting into schools, getting in very early where there's good genetic literacy in Americans broadly, so that people know that there is this access, that there is the possibility of doing genetic testing, and then have precision medicine as well. So I think that's a big barrier.
Mike. Hello? Okay. Yes, and I also wanted to add in terms of what's already available is that one of the key findings or appreciations of studying epigenetic mechanisms is that it's basically a way of our genome to interact with the environment, what we call the exposome, right? And so things like diet and exercise truly engages our endogenous pharmacies. We may not, in terms of cellular type and response, in terms of gene regulation and transcripts. And so these things we know are good for us: proper diet, exercise, reducing stress. This exposome effect on us is very much ways to intervene very cheaply. And just taking appropriate vitamins, supplements can indeed affect how our cells are responding, cellular diversity in terms of transcript responses. And so these things we could put in place outside of just more direct drugs.
Thank you. Middle.
Hi, or is it their turn? Or my turn? Go ahead.
Go ahead.
Okay, thank you. Hi. I'll go?
Yes.
Okay. Sorry about that. Hi, my name is Vasant Tharaja. I'm a second-year medical student from Turi University. It's actually my first AES meeting, so I'm really excited to be here. And it's been a really great set of presentations. I've been truly blown away. Like, all of the talks have deeply inspired me in my own research. And so I had a couple of questions for Dr. Rao about the neurostimulation devices. So you were talking about combining the use of different neurostimulation devices for treating patients with epilepsy. I just wanted to clarify, does that mean you can implant multiple devices, or do you substitute like one device for another in case the first device isn't working? And I'm also curious, what's like the primary deciding factor that you use to determine which device is most appropriate for a patient with epilepsy?
I'll take the first question first. That's a little more straightforward. Yes, I'm talking about implanting multiple devices in the same person, that you can do that. You can also explant devices. So if somebody doesn't want the device anymore, it can usually be removed. The second question is a bigger topic, which is how to choose devices. But the short answer is we often predicate it on intrinsic features of those devices. So, for example, if recording long-term data you think would be useful to you for any purpose, then RNS is the device that really has that feature in spades. If somebody is doesn't want to have brain surgery, then VNS is an extracranial device, and so brain surgery is not necessary, and so on. So it's usually intrinsic device features that help us on a per patient basis to figure out the right device.
Great, thank you.
My question is very generic on epigenetics. Since epigenetics research is coming up so fast, it's very difficult to keep up with that, number one. Number two, at least in oncology therapeutics, I think quite a bit of companies and agents are in clinical trials. So my question is just generic one, in terms of the considering the potential for in epilepsy therapy, so how do you think it will pan out in clinical research and clinical trials, and especially in terms of especially the formulation aspect of it? Is it intrathecal or intravenous or oral, assuming that some of the epigenetic agents are successfully going to have some efficacy in treating epilepsy?
So great observation that the field of epigenetics is fastly rising, and our understanding of the mechanisms involved are very complex. But we can learn a lot from the cancer literature in terms of how these epigenetic mechanisms, in terms of route administration of drugs and so forth. We're also learning that region specificity, cellular specificity matters in terms of the genetic phenotype of each cell type, and so targeting them more directly. So basically the development of new approaches for drug delivery is where we need to be in terms of manipulating these epigenetic mechanisms. Appropriately, and what we're learning is that the epigenetic landscape that is impacted, let's say with epilepsy or seizures, is very different than other neurological disorders. And so precision medicine and the precise way how these epigenetic mechanisms are altered in the context of epilepsy, these studies need to be done. And it's not just one approach is effective, right? There's different approaches and different ways to target. So viral loading or approaches to target specific cell types are necessary. How to get across the blood-brain barrier, that's also a barrier for us. And appreciating just in general how these epigenetic mechanisms are regulating gene transcription was necessary.
Thank you.
Thank you. We have time for two short questions. Go on the left.
Thank you. I'd like to echo the acclaim for the quality of the program first as well, and for the importance of kind of vision and creativity that the speakers showed in inspiring us. And for Dr. Rao, a lot of the features and biomarkers, the network-related biomarkers that you mentioned as potential predictors of stimulation efficacy, it struck me that there are also kind of components of seizure severity. You mentioned the synchronizability and the ability of the seizures themselves to kind of spread and recruit other networks. Is there does that impact on the relationship between seizure severity and responsiveness to stimulation?
That's a good question. Thanks for that question. I think it's a really good point, that severity and some of the network characteristics I described, could be related. There's not great evidence, to my knowledge, that seizure severity itself is predictive of outcomes. That, for example, very severe. How we define severity, by the way, which could be duration or type of seizure. It's not clear to me that there's data that necessarily correlates with outcome. In fact, sometimes we see with medications, for example, that it's easier to control convulsions than it is to suppress auras, for example. So I think that is something that we could explore over time. The challenge really is to quantify seizure severity, which is not so trivial to do. But that could be an answerable question with these devices. Is if you can quantify that, then you can look. I mean, most of the device trials have incorporated some severity scales of seizures and things, but I think we could probably do that in more detail as you're suggesting. Thank you.
Dr. Boston.
Thank you. Great talk. I just want to congratulate and thank each of you for a fantastic seminar this morning. And I just want to say that I hope Dr. Lowenstein's predictive abilities are better than they were with the Segway and the Google Lens, because I really do like, Dan, your current view of the future. So let's hope that comes true.
My question is for Dr. Hunt about inhibitory interneurons, and I think some of us are aware that Neurona is doing this kind of work, and 5 patients have had these inhibitory interneurons. Is your work related in any way to that clinical trial that's underway, and can you make any comments about the clinical trial?
Am I on?
Yeah, go ahead.
I'm on. Okay, indirectly, yes, the company was launched off my work at UCSF. I'm not involved in the company, and I don't have a comment on what they're doing. I simply don't know what they're doing.
Okay, thank you.
There's one there, and then maybe Sankar can take the last one. So go ahead.
So, first of all, Manisha, thank you for putting together such an amazing presidential session. My question is also for Dr. Lowenstein. So I noticed in your predictions, you left off the last part, which is, will we have neurologists actually enough? You know, we have a lot of APPs working with us. How are we going to manage that and make sure that neurologists are still part of the clinical program? You know, at least that's what, at our session, we have lots of APPs.
Yeah, Barbara, very important question. Who knows? But I personally have no doubt that the need for neurologists, the need for epileptologists, the need for expertise from a human is going to be absolutely essential for as long as I can imagine. And I think we have to keep in mind what the current disparity is in access to expertise. So I guess it's possible that, you know, the AI assistant will get to the point where it can provide much of the needed information, but there's never going to be a replacement for the human component of the interaction between a person who's suffering and needs the kind of expertise that only comes with a lifetime of experience. That's my own opinion.
There are robots that show that there's lots of empathy that they can display, and there is that sort of the AI that's also coming. But I hope you're right.
We'll see.
Last question from Dr. Sankar.
Thanks for the opportunity. I won't take much time, just want to make 2 things. Number one, kind of a question or a comment to Professor Rao. That is, of those various modalities, something that kind of strikes me that's not discussed enough in these comparisons is there have been a number of studies around the turn of the century by people like, you know, Andy Kanner and Frank Gilliam, focusing on quality of life in epilepsy, where it was found that would derive that whether your seizure reduction is 55% or 70% wouldn't really move the needle on quality of life. However, the importance of Kanner's work was the value of mitigating comorbidities. So my point is, perhaps there is added value to being in the brainstem and adding, you know, stimulating the raphe and the locus coeruleus. That may be bringing more to quality of life than is apparent, because I think at least I'm from UCLA and my residents are now very enamored of new technologies, and I'm sort of bringing this point that maybe the VNS is underrated a little bit. I don't know if you agree.
Yeah, I think it's a fair point, and I appreciate you bringing quality of life to the forefront. It's easy to focus on small differences in seizure frequency and things, but at the end of the day, it's really about quality of life. We and a group of us are actually putting together a new outcome scale to measure quality of life with neurostimulation devices for exactly that reason. I think VNS has its place, but I would point out that other technologies are also being studied almost as platform technologies to treat things like PTSD, anxiety, depression, obsessive compulsive disorder, lots of other sort of neuropsychiatric comorbidities, so chronic pain.
Thank you. The only other comment I wanted to make, just somewhere between Kelly's immediate problem and Dan's eventual solutions by 2050, I think we may be turning a little bit of a corner, maybe not a major point of inflection. I'm personally, as a medicinal chemist, formerly, very encouraged by the fact that there is a new category of drugs coming that I think are different than many look-alike drugs we have had. And going back to molecular biology and genetics, I like the fact that we have drugs that are emerging, and cenobamate is an example, where there's actually a selective blockade of NAV 1.6. So, you block the non-inactivating current, lowering the likelihood of APs, but it doesn't touch the APs, or it does not touch NAV 1.1. So the fast-spiking GABAergic interneurons are intact. So I think hitting excitation in the pyramidal cell without kneecapping the interneuron is a significant enough advance in my thinking, and there are even more selective compounds that are in the pipeline. And then, of course, there are potential for KCC2 enhancers. Just wanted to bring a little pharmacology.
Thank you.
Yeah.
Thank you very much. And please join me in thanking our outstanding panel of speakers. You've been a great audience, and thank you for coming to Orlando, and I hope you enjoyed this 77th annual meeting. It's been a great attendance, and thank you for staying late and finishing our discussion. Please claim your credit with the AES website and meeting app, and remember to attend the poster walking tours, which are listed on page 41.